so the theme of the symposium, I'll try to describe to you. So drug discovery and development as such, it's a very complex process, very high risk and high payoff process. And in order to find quick solutions, it's not easy through this approach. So drug repurposing, whatever the drugs we already have, it could be utilized. So Professor N. Srinivasan, chairman from Molecular Biophysics Unit of Indian Institute of Science, he'll speak about his work on drug repurposing and computational chemistry and medicinal chemistry, and how we can approach this problem of solving drug requirements. Dr. Rishi Jain is a medical doctor. He is a vice president of Wockhardt. He's going to bring in the recent developments in artificial intelligence for both drug discovery as well as developmental purposes. And Dr. C.N. Ramchand, who is our chief guest, Dr. and Professor Naidu, Dr. Balram Patro, Dr. Chandrasekhar Korpala, all of them will speak on various strategies each company is using for the process of drug discovery as well as repurposing purposes. Dr. Dinkar Sahal will start, will give a talk on inventing new drug combinations and also rational drug design, so that whether how far we can take it further. And again, same thing on similar theme. Dr. Nagendra Singh will also speak about various requirements of rational drug designing. Another important aspect, often it is ignored, it's about natural product research, Ayurvedic medicine, Siddha medicine. There's a lot of talk about it, but however, when it comes to real world, people have sense of doubt about it. So there are experts from these companies lifelong. They have been practicing these alternative medicine strategy for various diseases. It's really helpful, and they're going to bring in new approaches using modern chemistry approaches and biological approaches. They're going to talk to us, especially Dr. Narasimhan from Dr. Narasimhan, Dr. Ramesh Warrior, and Dr. Rajesh Kumar. They'll all speak about it. Dr. Ramchand will talk about repurposing drugs as well as reverse pharmacology. This is very important in the field. So I think this has been a very academic and industry interaction. We hope to get collaborative projects together and work together and solve the problem of this COVID-19, which is affecting all of us. Thank you all for your attention. I'd like to request Dr. Aisha to go further.
Thank you, sir, for giving a brief introduction regarding the symposium theme. Now, I would request Founder Director Professor M.A. Vijayalakshmi ma'am to introduce about the Center for Bioseparation Technology to the gathering.
Thank you, Dr. Aisha Noor. So it is a very great pleasure to be with all of you. Unfortunately, I'm not able to be there physically because I'm prisoner in Chennai. But I think I will give a brief description. Most of our participants and partners and speakers know about this center. So this center was created in 2005 with the separation technology as a core expertise, because that is the most important thing needed in any pharma industry. And with that, we started different aspects. Aisha, you yourself started with the natural product, which has gone a very far length and along in collaboration with different industry. Initially Ramchand, now Ramesh Warrior, everybody. So they are all our partners. And then I think the center has come up very well, and it has been promoted as advanced center for separation technology and related work, because we are doing all different multidisciplinary aspects. We have understood very early that the interdisciplinary approach is a very important with crosstalk between people. So we have also worked with chemistry department and a few other departments in VIT. So I think we have, in fact, we have a common patent with them, American patent. So all this was possible because of the team spirit that we had, and because of the fatherly, or I don't know what word to say, the support from my beloved Chancellor, Dr. G. Viswanathan, and his sons. I never had any problem with them. I chose Vellore as my choice to set up this center in 2005. But in the beginning, everybody, even the government, was doubtful how it's going to be. And they were very worried each day saying that, how is it? But I think kudos to VIT. I think this is the best. This is my honor to say that this has been a big success, and then it will continue. The road will continue because we have chosen Professor Krishnan as my successor director. I am very thankful to him. And he is keeping the team spirit that we have created. He is keeping that interdisciplinary aspect. And moreover, he is keeping very strong all the industry relation, in that sense that he has got even a project for translation of our patent, which we started in the beginning. I started, and he is now taking it to the product. So this is the way anywhere it should work. I think this has just been a model that the successor is able to really follow and then do things. And this is because, again, I will come back to my beloved Chancellor Viswanathan. He is able to maintain and encourage this kind of structures and this kind of people relation. He has never really created any problem in any way. So this people relation is nurtured by VIT. I think I should really thank them. And this nurturing is followed by his sons, who are really taking care of us in every aspect. So this is the occasion I want to put on record my most sincere, and I don't know how to feel, I have tears in my eyes, to thank them and ask and request them to continue this center forever, and we should see that flourishing and really helping the humanity. Thank you.
Thank you, ma'am, for giving a detailed introduction regarding about the Center for Bioseparation Technology to the gathering. Now, I would request respected Mr. Shankar Viswanathan, Vice President VIT Vellore, to address the gathering.
No, he is not there.
He's not there.
Now, I would request our chief guest, Dr. C.N. Ramchand, CEO, Saksin and Genome Technology Private Limited, Chennai, to address the gathering.
Good morning. Good morning to those who are in Zoom dais, those who are in India, those who are in abroad. I am telling abroad because, last time also, several people attended from overseas. So good morning to all, and Honorable Chancellor Professor G. Viswanathan. I understand Vice President Dr. Shankar Viswanathan is not there, but then let me come back to him too. Professor Vijayalakshmi, everybody calls her Professor Viji. And of course, the force behind this conference, Professor Krishnan, my collaborator too, and all the colleagues from academia, industry, and all the participants. I'm sure that several hundred or several thousand people are attending this, especially through YouTube also. So welcome to this conference. I'll be just briefly talking about a few things as well as about the conference theme also. And I've given about 10 minutes by Dr. Krishnan. So again, welcome. And Dr. Krishnan has already mentioned, we are in a different world now. Yes, that's right. The last 4 or 5 or 6 months that, many of the people have not even gone out of their house. So it is in a different setup. No restaurant is working, no institution, no universities. So altogether a different situation. The only difference is that compared to the pandemic of 2018, there is certain difference also is there. So welcome to everybody for this great conference, second in series. The first Dr. Krishnan organized was on the diagnostics and therapeutics, which is connected to COVID-19. And today it is going to be the drug discovery, development, drug repurposing, then immune potentiation, and reverse pharmacology approach. So various different approaches that will be covered in today's conference, 2-day conference. Almost about 12 people are going to attend. Before going into that, let me briefly tell about what I understand from about VIT. And before I know VIT very well, I knew Professor Vijayalakshmi. We met about 10 years back at a winter, maybe 15 years back in a marriage, and we had several interactions since then. So I will briefly tell about that little later. And let me also tell about VIT a little bit. I am sure that I don't need to introduce, tell, or, all about VIT because everyone knows about VIT. But what I understand is that, VIT is one of the major players in higher education, not only in India, internationally. I've also seen that one of the best universities in the world, one among several thousand universities. That's a great achievement. In few years' time, some university can reach up to that level. And it is a high-tech institution. An example is CBST itself, engineering faculties, lot of research papers, lot of patents. And it has won several awards, including ARIIA and FICCA. ARIIA is probably the evaluating agent by the Human Resources Ministry. So these are the types of achievements that VIT has got. And recently it has been recognized as an institute of eminent status to go up its stature in global ranking of education institutions. So I'm sure I don't need to tell too much about Bengaluru Institute of Technology, its achievements, but certainly I would like to briefly tell about your chancellor, or let's say our chancellor, one of the most well-known chancellors in India. I don't know, Professor Vishwanath, you remember me or not? I have met you 5 times, twice with the Professor Vijayalakshmi, once with Professor Ajay Kapoor, and we also met in Tamil Center meeting in Chicago. So I came and met with you. Of course, you are very, very busy. So I had such a fantastic interaction with you. That was the very first time with Professor Vijayalakshmi. We spent quite a lot of time, and with Professor Ajay Kapoor too. So it's such a good vibrations, like, positive vibes you get when you talk to him. And yesterday I was talking to Krishnan, and he had amazing words to tell about the Chancellor. His energy, he's young, looks young. So I am sure that several decades to go, and many more institutions, not only Bengaluru Institute of Technology at Bengaluru campus, Chennai campus, Bhopal campus, many other places. I'm sure that you will enter into internationally other places also. So it was a great pleasure meeting you many times. I hope to meet with you many more times. Now, let me also tell you about brief about CBST, Professor Vijayalakshmi, and Professor Krishnan. Now, I have been associated with both CBST and Bengaluru Institute of Technology for the last 10, 12 years. I started with few conferences. Let me tell you, Professor Vijayalakshmi really helped in setting up a molecular medicine conference, and subsequently we conducted 2 molecular medicine conferences. I have been few times as examiner for PhD as well as MTech theses. I came many times to teach as faculty for or small conferences which Dr. Krishnan or Professor Vijayalakshmi has arranged. And CBST is closely working with industry to develop the products with support both Government of India and industry through Make in India under National Biopharma Mission by BIRAC Government of India. An example, Dr. Krishnan got few projects under the BIRAC program, mainly connected to COVID diagnostics. And the most important thing is that 3 people are really involved in developing this Center for Bioseparation Technology. And let me also tell you, the one of the unique institutions anybody can see, whether in India or internationally. There are many, many different institutions out there. But Bioseparation Institute, I have not seen many places. But let me tell you, bioseparation is one of the most important part of any drug discovery development program, whether it is small molecules, biologics, or alternate applications of drugs or vaccines. Separation, that means without impurity. And everyone fails the most failure in industry is because of the problem associated with the biosuppression. And it's great that Professor Vijayalakshmi set up this facility in India, especially in Bengaluru Institute of Technology. That's why she could produce several PhDs, several collaborations, etc. And I'm really great that I'm in touch with Professor Vijayalakshmi. Great inspiration, not only for BIT, not only for CBST, many institutions, because when I go to IIT also, I hear about that, especially some of the new things what she is setting up. I don't want to tell you her age, but then she works like a 30-year-old person. Lot of energy, surrounded by whether youngsters or elders, everybody will get energy to do that. I'm sure Dr. Krishna also has got that energy to continue with that, the precedence of whatever she has been doing. So it's great to be in touch with all of you. Let me also now come back briefly about the conference itself, the theme. I hope, Dr. Krishna, I have a few more minutes. Right. So
Yeah.
Dr. Krishnan has decided a very apt, title, and the people who are involved in talking mainly from industry also, those who have involved in developing drugs, biologics, or other products. One of the things he has mentioned is that the challenges, opportunities. So if you see, there is a great challenge now. So let me read out the title. So it is basically disease control, opportunities and challenges for drug discovery, development, drug repurposing, and alternative medicine. So this reasonably covers most of the things. Now, you know about disease control. Really, we do not know how this can be controlled. It's all hypothesis based. But we know that, social distancing might help. Then later it came that probably that's not going to help. The reason is that because, the virus can spread through the atmosphere also. But earlier that was not the thing. Earlier it was 1 meter distance. Now it is 2 meter distance. Second thing is that, everybody told to cover with a mask. That also came as a problem. Sometimes people are telling mask itself is going to be a problem because many people are mainly touching here and there. But then do not worry about that. Please see that everybody keeps social distance and a mask, and if it is possible to confine to your house, it should be there. Now, what are the opportunities? Drug discovery and development itself is a great opportunity. If you really see the drug discovery and development, we have still to achieve a lot. You see about 35,000 diseases, only few hundred diseases really has got drugs. Now, the opportunity is tremendous. Now, if we just see about viral disease, there are 270 viruses are there for simple cold. Everyone knows that there is no medicine for cold. The problem is that, first of all, the viral genome is continuously changing. You don't know, maybe 50 years back, what was happened with the specific cold, that may not be the one. It's constantly increasing, changing. That is one of the reasons. It's not easy compared to other, for example, if it is bacteria or even human targets, it's comparatively, of course, difficult. But if you talk about virus, it's much more difficult. The reason is that virus has got only few proteins. Many of them are structural proteins. Functional protein like, RNA-dependent RNA polymerase, DNA-dependent RNA polymerase, DNA-dependent DNA polymerase, certain protease, integrases. Only few molecules are there. On the other hand, the surface of the protein, the virus has got many other structural proteins are there. So that might be good for developing vaccines or antibodies. But if you are developing small drug, it has to be a functional protein. And for example, only 5 or 6 proteins are available. That is one of the reasons. And suddenly a protein comes up. It is not easy to develop a drug against that because, drug discovery is a 15-year process. Very complex. So that's why you might have been heard about drug repurposing. So currently, that's if you want to go about small molecules, that's the only possibility. That's why at least 4 or 5 different molecules have come. Probably that can be used. On the other hand, the second possibility is that You will be able to develop antibodies, antibodies against the surface proteins, antibodies against certain other structural proteins, or you will be able to develop attenuated virus or killed virus. This can be used, or RNA vaccines, DNA vaccines. So these are the possibilities. But luckily, if you see that that 15 years and everything, in 2 or 3 years time, things have started coming. Maybe 1 or 2 years already, vaccine test has started. Now that means if you really want, that can be done, because the development took place in few months' time, and immediately that the regulatory compliance, the people involved suddenly started giving all the approvals. That's a great thing to happen. Then you heard about, I briefly talked about drug repurposing. Drug repurposing is the existing drugs, which is only 420 drugs are there. Out of that, maybe 10, 20, 30, mainly viral drugs or other small molecules can be used to develop as viral replication inhibitors. So that is one possibility. Now, there are other possibilities too. There are many failed drugs are there. So people are trying on that one. So I'm sure that the first talk is going to be that. Failed drugs in terms of efficacy, but which do not have toxicity, which do not have any pharmacokinetics problem. Those are other drugs that can be done. The other one, people are talking about immune potentiation. Very important. Immunity, how you can improve immunity. Let me tell you, there is no clear cut or, the right drugs, molecules is not there. I'm not telling about the innate immunity or barrier functions, which we can control. But if you are talking about the adaptive immunity, how to increase that one, really there is no proper drugs has been done. I'm not telling about the adjuvant therapy, adjuvants for vaccines. That's a different thing. So this is some area. For example, how the efficacy, effect of T cells can be improved. T helper cells or cytotoxic cells. So this is another one. And finally, that alternate applications of medicine, alternate applications like, let's say, Ayurvedic medicine. The problem is that if you really see only few, of course, originally most of the drugs has come from natural product, but you have to remember one thing, many of these has been improved. That is, structural modification has been done. But in the last 15, 20 years, nothing much has come. So, but there is an opportunity there. What we have to do is that there is something called reverse pharmacology approach and see whether something can be developed. So these are the things that will be covered. And thank you very much. Thank you, Dr. Krishnan, especially for doing this great thing. And one over the other 2 conferences you are organizing, and especially thanking everyone for making me the chief guest and to give this talk. And I'll be talking about my talk about all about drug discovery, biologics discovery, alternate applications of drugs, and immune potentiation, etc. tomorrow. So thank you very much. Thanks. Thank you.
Thank you, sir, for sharing your thoughts about briefly about symposium themes, about CBST and VIT. I request respected Honorable Chancellor, Dr. G. Viswanathan, sir, VIT, to deliver his presidential address.
Good morning. Our chief guest of today, Dr. Ramchand, Dr. Vijayalakshmi, the founder director, Dr. Krishnan, director of the Center for Bioseparation Technology, Dr. Jayaprakash, Dr. Aisha Noor, and other professors, the various speakers from all parts of India and globe, dear students, participants, very good morning to you. The topic chosen for this 2 days conference, symposium, is COVID-19 disease control opportunities and challenges for drug discovery and development, drug repurposing, and alternative medicine. This is a very important subject today, because the disease has affected all the countries of the world. And we have invited eminent speakers. I would like to thank all of them, like Professor Srinivasan, Dr. Rishi Jain, Dr. Balram Petro, Dr. M.U.R. Naidu, Dr. Chandrasekhar, Dr. Dinkar Sahal, Singh, Dr. Ramesh Warrior, Dr. Narasimhan, and Dr. Ramasamy and other speakers. I'm happy that the chief guest and other professors have accepted our invitation to be here for this 2 days symposium. This center was started in 2005 by Professor Vijayalakshmi. I don't know how many of you had the opportunity to move closely with her. In fact, I visited her laboratory in France even before her return to India. And she was yet to finalize a place where she is going to set up the laboratory center. And finally, she chose Vellore. And even though she is formally retired, she continues to be a professor here and visits often, except this lockdown period. And she is not only a teacher, she is a philanthropist. In fact, she has contributed lakhs of rupees for the poor children's education, where we have a trust called Universal Higher Education Trust in this district. We have about 740 districts in the country. And this is the only district where we are helping children to go for higher education without any government help. Almost 5,000, more than 5,000 students have been given this facility. And she is one among the major contributors. Of course, VIT contributes about 60% of the collection, and the rest is contributed by the public. And she is one among them. In fact, people used to ask me, how are you able to get along with her? She is a very tough lady. I used to tell them, if you understand her, then it's not a problem at all. Sometimes she will be aggressive, sometimes she will be submissive. She is always a permanent student, goes on learning, goes on studying. And I found her enthusiasm in bringing many things, many instruments from France, not easy, because something like bringing Sridhana property. She brought things from France. And of course, I would like to thank Government of India for supporting this project. And they were generous in supporting this. And now it is doing very well. I congratulate Dr. Krishnan and all his colleagues for making this a wonderful institution in the center. I thought I should introduce VIT. I would like to thank Dr. Ramchand. He has introduced almost half of it, or more than half, because we started in 1984 as a small college called Vellore Engineering College, affiliated to the University of Madras. And our student strength was 180. In 2001, we were given a university status when Atal Bihari Vajpayee was the Prime Minister. And we started growing. Now we have 4 campuses: Vellore, Chennai, Amaravati, Andhra Pradesh, and Bhopal. We have 46,000 students now. And as the chief guest pointed out, we have become one of the best universities in the country. That's why they have chosen us as an institution of eminence, IOE institution, which has been given the status for 20 institutions in the country. We have students from all parts of India, all the states and union territories of India, and also from about 60 countries of the world. VIT provides topmost priority for quality education and focuses on research and development, which is reflected in terms of research output with respect to number of publications and patents. In the last 5 years, we have become number 1 in the number of Scopus Index publications. To begin with, we were number 2 behind the Institution of Science, Bangalore. Now we have overtaken them also in number of publications. And of course, we have to improve quality. And I would like to thank Government of India for recognizing VIT as an institution of eminence. This will help us to compete at the world level, to move in the world ranking of educational institutions and universities. Our goal is to become one of the top 200 universities in the world, attracting the best talent, nurture them, and develop human resources to serve in knowledge-intensive economies. My belief is that India should lead the world in health, wealth, peace, and prosperity. I always tell our students, when India leads the world, VIT should lead India. I hope we will be able to achieve it. Health and wealth of a country depends on the development of cutting-edge technologies and services which will be useful to mankind, and thus VIT is focusing on international research to meet such demands on mankind. Professor Vijayalakshmi, a pioneer in bioseparation technology, who has spent more than 40 years in France, has started this center, as I told you, with the financial support from Department of Science and Technology, Government of India. The center mainly focuses on the oriented research by integrating bench to bedside concept and bridging the gap between academia and industry. Her successor, Professor Krishnan, following her path, I'm very happy to inform you that all our faculty members from this center was able to get a sponsored research project from Department of Biotechnology, BRAC, to develop immunologist kit for COVID-19 infection. Soon our faculty members will fetch more such competing grants to find solutions to tackle this pandemic by developing therapeutics, vaccines, drugs, and also use our traditional knowledge of herbal medicine, either for treatment or for a cure. This can be done only with the help of collaboration between academia and industry. This conference helps us to bridge the gap by promoting academia and industry interaction. Two important aspects such as cost effectiveness and affordability should be prioritized while developing the new drugs or therapeutics to better serve the wide spectrum of people. I'm very happy to see that the scientists from both academia and Indian pharma industry have agreed to come to this platform and share their knowledge and experience with students and delegates of this symposium. This will serve as an inspiration to young minds to succeed in this knowledge-driven endeavors. This meeting will discuss new drug discovery program, repurposing of already existing drugs in the market, and potential traditional medicines and reverse pharmacology for treating COVID-19 infection. This is extremely important as our country has made rapid progress in both pharma and biopharma industry, and we need to capitalize on the momentum to find quick solutions for the benefit of our society. This symposium will be attended by more than 900 students and young scientists across India and participants from USA, Malaysia, and few African countries through virtual platform. I take this opportunity to thank each one of you for coming to the symposium, and I am sure you will reap the benefits upon interacting with scientists of academia and industry who have come from different parts of the country. I'm very optimistic that you will get an opportunity very soon to come to our campus located in different parts of the country and interact with students and faculty, exchange ideas. I would like to congratulate our faculty, Dr. Krishnan and others, and our students of this conference for bringing eminent scientists from prestigious academic institutions and leading the pharma industry to this virtual platform. I wish this symposium to be a grand success and wish you all a safe and happy life. Thank you.
Thank you, sir, for your encouraging words and sharing your valuable thoughts. We have now come to end of our inauguration session. I call upon my colleague, Dr. N.S. Jaiprakash, Associate Professor Senior, CBST, to deliver the vote of thanks.
Yeah, thank you, Dr. Aisha. A very good morning to all. I consider it a great privilege to propose a vote of thanks to all the dignitaries, scientists, students who have made it convenient to attend this inaugural session of this virtual symposium on COVID-19 disease control, opportunities and challenges for drug discovery and development. At the outset, I would like to thank our chief guest, Dr. C. N. Ramchand, CEO, Saksin Life Sciences Private Limited, Chennai, for his esteemed presence and gracing this occasion in spite of his hectic schedule. His presence today in this function has immensely enhanced its importance. Thank you, sir. And we are deeply grateful to our Honorable Chancellor, Dr. G. Vishwanathan, for his august presence and presiding over this function. So thank you for your enlightening address. And we are honored by your esteemed presence, sir. Yeah. I take this opportunity to thank Mr. Shankar Vishwanathan, Vice President, BET Bellu, and Assistant Vice President, Ms. Kadambari S. Vishwanathan, for giving us constant support and help in all our endeavors. I thank our Vice Chancellor, Pro Vice Chancellor for their profound support for conducting this virtual symposium. I must express my deep gratitude to our Professor Vijay Lakshmi, our Founder Director, CBST, for her initiative and guidance to us all in our activities, ensuring that we always do the right thing in the right way at the right time. Thank you, ma'am. I thank Professor Krishnan, Director, CBST, for all the initiative he took in holding the symposium and also coordinating and inviting the eminent speakers. Thank you, Professor Krishnan. I also express my sincere thanks to all my colleagues and especially the students at CBST for their enormous cooperation in organizing this event. A special thanks go to Ms. Neha Deoraki, a PhD student CBST, for preparing and collating all the information much required for this program. Our sincere thanks go to Dr. Kalikishore Reddy and Dr. Sanjit Kumar, CBST, for helping us in the technical issues of this particular Zoom software platform. Thank you. Last but not the least, my sincere thanks to all our participants who have taken keen interest in the program and have taken time out of their busy schedule to attend this symposium. Once again, I thank you all. Thank you all.
Thank you, Jay. Now I request our director, Dr. Krishnan V, to chair the first technical session of the symposium.
Thank you. Thank you, sir. Thank you so much. Thank you.
Thank you, sir.
So I need 1 minute to start the PowerPoint presentation to introduce this first speaker.
Okay.
Thank you, sir. You happened here, huh?
No.
Is it visible? Hello? Hello?
Yes, Dr. Krishnan. Dr. Krishnan, it is visible. Yes, yeah.
Okay. So we take just 10 seconds, 20 seconds break, and then we start. I hope Professor Srinivasan is ready. So I will introduce Professor Srinivasan, who is going to be our inaugural speaker. Professor N. Srinivasan is currently a professor and chair of the Molecular Biophysics Unit at Indian Institute of Science, Bangalore. And so he's leading a research group in the area of computational biology and bioinformatics. He's published more than 310 scientific articles and of international repute. And he's leading a very big team in IISc Bangalore. So he has won several awards and fellowships at both at national level and international level. So he's a recipient of National Bioscience Award. He's a recipient of Shanti Swarup Bhatnagar Award, which is a prestigious one. Fellow of the Indian Academy of Sciences, Fellow of the National Academy of Sciences, and of course, J.C. Bose National Fellow. At international level, he is a Senior Fellow of the Wellcome Trust from London, UK, Senior Fellow of the University of Manchester. He was a visiting professor for bioinformatics at Union University and Nantes University in France. He serves in many journal editorial boards on bioinformatics and biology direct, etc. So he's going to talk to us about. So I think he's one of the first recipient of this COVID-19 grant from BIRAC. So he's going to talk to us about repurposing drugs against the main protease of SARS-CoV-2, mechanism-based insights supported by available laboratory and clinical data. Over to Professor Srinivasan. Thank you so much for accepting our invitation. The podium is for you. Thank you.
Thank you. Thank you very much, Dr. Krishnan. And I will try and share my screen. Okay. Just a minute, please. Are you all able to see my first slide in full screen?
Sir, yes, sir, it is visible, sir.
Okay, fine.
You can make it a full screen, sir.
All right. Okay. Yeah. Thank you. Thank you very, very much for a very kind introduction, Professor Krishnan. And I also would like to thank all the authorities of the VIT for inviting me to this conference. I have been having a very friendly relationship with VIT for many, many years. Maybe more than about 12 years, I would think now. I've been there many times as the examiner of PhD thesis on some various, the committees. Moreover, until about 3 years ago, my daughter was a student in VIT. So I have a very fond memory and attachment to the VIT, both at the professional and at the personal levels. And it's very nice of VIT to invite me, and I'm very happy to be back, at least in a virtual fashion, in the VIT. Okay. At the outset, I have to make certain confessions. First of all, I'm not a virologist. I'm not a specialist in the pharmacology. I don't even have a formal background in biology. So my research interests are over the last 30 and odd years is to try and understand the complex relationship between amino acid sequences, three-dimensional structures of proteins, their functional properties, and their interaction properties. And this has been my bread and butter for a very long time. But this is such a broad theme, it can have implications on diverse fields. For example, in the signal transduction, in the understanding of metabolic pathways, and in the protein folding problem. And one of those interests, it so happened that in the context of repurposing drugs. So I am not really a specialist in this area. And secondly, I have to disagree with a statement made by my introducer. I don't have a grant to work on COVID-19. I tried something, and I think the committee felt that it can't be funded. So that doesn't stop me from working on COVID-19 anyway. Even somebody like Michael Levitt, who won the Nobel Prize a few months ago, before he got the Nobel Prize, we had invited him to visit Institute of Science for a conference to commemorate the contribution of Professor Jain Ramchandran. And then he had very kindly agreed, because he said, one of his heroes is Ramchandran. But just about a month before the conference, he sent a mail in a depressed tone that the 3 grant applications, none of them were successful, and only to win the Nobel Prize few months down the line. So the decisions of funding bodies doesn't matter to people if they are passionate about science. So okay, so on that note, I would like to, although we do many things in my group, I would like to talk about on this SARS-CoV-2. I never dreamt that I will be working on this until about early March. In fact, I never I never even, it didn't even come in my dream that I can make a contribution here. I was invited by a very nice college in Coimbatore in early March, and I was there to present my work on some recent analysis on the Ramachandran principles. And then I got a call almost in the middle of the night from my student, Shohini Chakrabarti, rather excited about something to do with the main protease crystal structure that is deposited in Protein Data Bank. She's a PhD student, one of the bright students I've had, who is looking into, among many things, on the accuracy of the crystal structures of protein small molecule complexes. And she has a rather rather interesting conclusion that many of the structures are not really reliable, almost like 80% of the structures. And she was, somebody was racing with time and to deposit structures on the SARS-CoV-2 main protease apparently bound to various inhibitor molecules. And she was quite excited to see, and she was also very concerned that many people will start using these structures. And if the structures are not reliable and accurate enough, it's all going to fall apart. It doesn't serve the purpose. And then she accidentally, rather serendipitously, began to wonder if the proposed bound complexes are not satisfactory, then what should be the bound complex? So it started rather as a curiosity to identify what could be the correct mode of binding of a small molecule to one of the proteins which turned out to be an attractive target of the SARS-CoV-2. So neither my student nor myself ever planned to get into this area at all. We were doing something else in which we were, pulled like a magnet into this. And so that's actually very, very nice because this, as told by the chief guest, the drug discovery is a very long process. One thing is very, very clear for those who are working on drug discovery: you have 99% chance of being failed in the drug discovery. So the chances of success is extremely bleak. And now, and again, it takes many, many years. So now the corona is knocking at our doors and killing people already. So it is, one of the ways to circumvent the problem of long time and a very, very bad rate of being successful in drug discovery is that many people, whoever is able to contribute in this area, should work together. And what Rosalind Franklin once told when she looked at the double helical structure of DNA worked out by Crick and Rich, that she said that your model looks correct from whichever angle I look at it. It's not as though she's looking at the model from the top or from the left or from the right. When she thinks about transcription, it makes sense. When she thinks about the duplication, it makes sense. And when she thinks about it's a genetic material, it is a carrier of genetic information, it makes sense. So if many people are working on this, and if using different computational and experimental approaches, if different people employing completely different approaches, both in terms of basic principle and assumptions, and and also in terms of technique that is used, if they end up focusing, if they end up coming with the same answer, for example, then you can probably have a high reliability on such a detection as a potential drug against the COVID-19. So one of the ways to circumvent the problem of, not circumvent, to minimize the problem of long time and bad success rate is that everybody who believes that he or she can make a valuable contribution should make a valuable contribution. So thankfully we got sucked into this matter, and now in retrospect I believe that it is nice that we are sucked into this matter. Having said this, we have been working on so-called repurposing drugs at least for the last 5 years. in fact, much longer. Here is a selection of publications we have we have come up from our past work. I will mention some of this as I go by. The last publication in this list that's shown in a different color is the work I'm going to be talking about. I don't usually submit my research article into these archives, but this is one of those things I felt it's important to deposit it in archives because if it is thrown open to the world, it is only a computational work. I mean, somebody might like to take it up to the laboratory or even the clinical level. So that's the only reason why we put it up in the archives, to make it open for anybody in the world to probe into this. But parallelly, we submit it to a Royal Society of Chemistry journal, Molecular Omics, and it is accepted. It is just yesterday, it is online. So I'm going to be only talking about very briefly about our work. The details are in the manuscript, in our paper. So, what is the basic principle about drug repurposing? On your left, you see some number of locks and you see one key. It is always possible to think of some sort of a key which can seamlessly open some the diverse looking locks. I will let you a little secret that I have learned over many years. I used to have the Samsonite suitcase with me, which I used to travel sometimes when I go abroad, a big one. And then I lost its key once. And then somebody in my family told that, never mind, any Samsonite suitcase key will open your Samsonite suitcase as well. I didn't know that. And she was absolutely right. And so I hope the Samsonite has woke up to it now, and they are making different locks now, not to compromise on the security of people using the Samsonite suitcase. So that's quite possible. So similarly, on your right, you see a little diamond in sort of golden color, which is an indicative of a molecule, a small molecule that might bind to a protein, which are shown cartoonistically at the top in different colors, gray, red, violet, and blue. And you see the apparent binding site there, which is shown like a V-shaped. It can take this, the diamond-shaped molecule. So it's possible that a single molecule can bind to the diverse proteins. If this is an inhibitor of an enzyme, for example, and this could be a problem. Usually, the off-target binding is something. Suppose you are targeting a pathogen which is residing in the human host, and then you come up with a molecule that will target the pathogenic protein and abrogating its replication or something. What in case it binds to a human protein? As it abrogates the replication of the pathogen, it might also kill the human. So that's a side effect. So it can work in the negative way as well. However, we can also use the problem of the binding to multiple targets in a positive fashion, and that's what is done in drug repurposing. In drug repurposing, what one does is that you have many drugs which are either used in the market. You can buy it across the counter in a pharmacy, or it has been tried for some disease, and for some reason it could have been blocked. Now it's not usable for that disease. But this was also mentioned by the chief guest in his speech at the beginning. So the problem in the drug discovery of the brand new molecule is that, as told at the beginning of this session, it has to pass through many conditions. For example, the side effect, the toxicology tests, all kinds of, before it comes to even to the level of the clinical trial phase 1, it is much more than the 36th chamber of Scholin, if you like. And who is that hero that will pass all these 36 chambers is, it is almost nobody at the end. So that is the fate of brand new molecule. But if already some molecules are proposed against certain problems, for example, an antibacterial, and suppose it is targeting, for example, it is targeting a protease, for example. And if you have the homologous protease or another target in a different pathogen, then it has a chance of binding into that as well. So what we call a side effect, if it binds to the host proteins, we just exploit it in a positive way. If that, the binding with additional proteins is actually it is coming from a different pathogen, which is our target. So the graph here shows it gives an idea about the benefits in terms of time and money that we can enjoy, that we can get by simply repositioning or repurposing the currently existing drugs. So actually, this is not new. There's a wonderful article published in Nature Review in 2016. Those who are interested can go back and read this. This is titled "The New Tricks for Old Drugs." This has been going on since a very long time. There are many examples, but I'm going to just highlight a couple of examples here. This is the story of the thalidomide, and another one is the metformin. The thalidomide is a drug which is originally approved by FDA for administering to the pregnant woman to avoid the morning sickness, and it was used by them also for it is mainly for the morning sickness. And unfortunately, the babies born to those women, they have deformed limbs, like you see very badly formed limbs here for these children. Their mothers, when they were in womb, their mothers took the thalidomide, and it is unfortunate. And therefore, once this was realized, then it was stopped. The administration of thalidomide has been stopped for the pregnant woman. But later on, it has been realized that it works against certain kinds of cancer. For example, the multiple myeloma, and also for certain skin problems like the leprosy. So it is almost, it's not yet fully approved, but it is a new hope that it could be used for certain kinds of cancer and against the leprosy and other skin problems. Another example that I have here is the metformin, which is originally an anti-diabetic molecule. And subsequently, it was realized that it is a good anti-breast cancer molecule as well, but still it is not approved by FDA for use against the breast cancer. However, it is well on its way. It is one of those heroes that might emerge out of the 36th chamber of Sholay, quite possibly. But the thalidomide is back with a fresh use for use against certain kinds of cancer and leprosy, but it cannot be administered to the pregnant woman. So this has been going on. There are innumerable examples like this. And so what we are trying to do is only just to use those old principles to see for the modern those unfortunate problems, can we use these at least. So this is what we have been trying to do. It's only a part of my lab activity. Here I'm showing 3 popular pathogens which have been addressed by our group in the last few years: the Plasmodium falciparum, the malarial parasite, Mycobacterium tuberculosis, the TB, and then the Candida albicans, which is causing certain fungal diseases. So we have proposed a number of molecules using computational approach to repurpose the existing drugs. And it's also nice to see that many of the drugs that we proposed in the past are actually being tested to work reasonably well. using the laboratory and the clinical studies. So we did not spend any time in choosing a target for the SARS-CoV-2. As I said, we never meant to get into it at all because we noticed apparent problems with crystal structures of this protein, SARS-CoV-2 main protease, as they are reported, some of the structures in the early March in Protein Data Bank. We began looking into this from that angle. Then we realized, oh, maybe perhaps what we are doing will be of general interest given the current situation. So what you are seeing here is a three-dimensional structure which is deduced by X-ray crystallography of the main protease of SARS-CoV-2. And this is actually, in retrospect, I feel this is a good target because as told by Professor Vijay Lakshmi, these viruses, they mutate a lot. And also by the chief guest. These viruses, they mutate a lot, and there are many mutations noticed in the SARS-CoV-2 proteins. However, the main protease is one of those stubborn cases where the mutations are not seen very much, at least as of May, a couple of months ago. I don't know whether anything new has come up on the main protease. So therefore, it's an attractive target. It is an enzyme which is a cysteine proteinase. However, it has a fold of trypsin, which is a serine protease. So we have actually all the work I'm presenting is almost all of it was done by my student Shohini Chakrabarti, who got into his rather unplanned, and she has employed two approaches, and one in the left is using the concept of structural neighbors of proteins, and which is exploiting the protein space, if you like, and then on the right is exploiting the chemical space coming from the small molecule side. I will explain what these are in the next slides. So so-called the workflow one, as Shohini calls it, it is expanding, it is enriching the protein space, if you like. So here is our target in the left top corner, structure of the main protease of SARS-CoV-2. And then we ask, I want to avoid these jargons, use of these jargons, and I will just say what scientifically we are trying to do here. According to this workflow one, we simply asked a question, what are the other proteins whose three-dimensional structures are similar to that of SARS-CoV-2? The interesting thing about protein 3D structures is that they have very limited repertoire. There are millions and millions of sequences, but the nature can't afford to give different three-dimensional structures to these sequences. Necessarily, the completely different proteins from completely different organisms have to necessarily share their structure. Mother Nature has to ration the protein folds because protein folds have to be adopted with the satisfaction of many chemical and physical grammars and their frameworks, tight frameworks. For example, hydrophobic residues should be inside. You can't have knots in the backbone of the polypeptide. So there may be many proteins with the same sort of three-dimensional fold as the main protease. And the good thing about identifying proteins with the same sort of fold as the main protease is that they may have very similar function, same or similar function, because the functions of proteins are highly governed by how they look in 3 dimensions. If you alter 3-dimensional structure, you have a potential danger of altering or abrogating the function of a protein. So the structure is really the central point to look at in order to understand why a protein does what it does. So we got large number of substantial number of proteins which show the 3-dimensional structural similarity of the with the SARS-CoV-2 main protease, and we have to bring in a lot of filters. I will cut the long story short, and then finally we came up to a small data set of 51 proteins because we want to be very careful about the resolution of structures and things. And then we asked a question: how many of those 51 proteins with known 3-dimensional structure has is known to be a target to a small molecule which is present in the drug bank. You have the drug bank in which all the known drugs are deposited, their details, various details are deposited about various drugs, and the targets of these drugs are also documented in the drug bank. So we asked a question, how many of the relatives, the cousins, brothers and sisters of the SARS-CoV-2 main protease are targets to the molecules in the drug bank? And then we came up with 51 proteins with 26 compounds, which are known to target 51 proteins. Some of the 51 proteins. And then all that we do, I'll cut this, and then we just consider these compounds, whether if they can bind to the cousin of SARS-CoV-2, can it also bind to the SARS-CoV-2 itself? Because the functional sites in proteins are conserved during the divergent evolution of proteins. That we have learned over many, many years of work. So if a molecule, if an inhibitor is binding at the enzyme active site, it's of one enzyme, it is possible that it can bind to the active site, the equivalent active site of an orthologous enzyme from another organism. So unless the inhibitor binds at a non-functional site, causing some allosteric effects at the functional site. So, we have, once you identify this, you have the 26 compounds free for you to investigate against binding to the SARS-CoV-2 main protease. It's quite logical and simple things that we did, nothing very fascinating. Nothing very intricate here. So, these are some of the molecules, 17 molecules, which are all either approved or at the verge of being approved. Okay, investigational. Those which are shown in the shining green color are already shown by either the clinical or the laboratory experiments as antiviral and specifically anti-COVID-19. They don't know what is the target. We know what is the target for at least one of the targets, which is main protease. And then the one shown in blue is very much in investigational level. Others are not yet explored either clinically or in the wet lab, as far as we can tell you. And so all these molecules which are picked up from the relatives of the main protease or the close analogs of those, we have to make sure that they could bind to the binding site of the main protease. And that's what is shown here. We have to use, this is a separate seminar by itself. We have to use the fundamental principles of physics and chemistry in order to assess the structural and chemical compatibility of small molecules at the functional site of the main protease. And you have, please believe me that all of these are shown to bind with very good energy values. And this is another approach which Sohini employed, which is coming from the small molecule, from the drug side. So what she asked here is that there are many known compounds, known inhibitors of SARS-CoV-2, as shown in the left top here. And then she's asking the question, just like the protein-centric approach in the workflow 1, in the workflow 2, she's asking the question, what are the molecules in the drug bank that are chemically and structurally similar to those initially proposed inhibitors of SARS-CoV-2 main proteinase? And then she gets a large number of compounds, and then she clusters them on the basis of their chemical similarity, and lots of scrutiny we have to go through. I don't have time to go into the details. Everything is in our paper. Those who are interested can take a look at our paper. And then finally, she performs docking simulations, and she came up with 18 other compounds from enriching the chemical space associated with the main proteinase of SARS-CoV-2. And these are all kinds of molecules, antibacterial, antifungal, and many others. Here is a list of those 12 promising molecules which are picked up using using the expansion of the chemical space, and most of them are have already shown to have some potential for antiviral. And you can see in the column number 3, the status, either many of them are approved or investigational. So the things are looking very promising from our data. And so again, we have to ensure that all the energetics of binding are all fine. So the key point so far is that based on our analysis, we prioritized 29, 17 from protein-centric approach, 12 from small molecule-centric approach, 29 molecules which are the top set from our analysis. And 20 out of 29 are approved by the FDA for treating into some problem or other, not yet for against the COVID-19. There could be some other viral, antivirals or antibacterial, etc. The remaining 9 are investigational drugs. Apart from 29, there are also 125 drugs which are not, which are not at the topmost level in our ranking as the top 29. And it is also very nice to see that many of the proposed molecules are already well on their way. As the exploration against the COVID-19, for example, the remdesivir, and then the ribavirin, etc. And then it is also very nice that although some molecules are shown to be having some anti-COVID-19 activity, people are not aware of what the target is. So the improvement on those inhibitors would be very difficult using rational drug design approach if you don't know what the target is. Our work also suggests for some of those exploratory inhibitors, the target is domain protease. And there are also many other molecules which are proposed for the first time, and so far we are not aware of any laboratory or clinical exploration of these compounds. And another interesting thing is that some of those drugs, some of those inhibitors, I should be cautious. Some of those inhibitors, they are not yet shown as drugs, so I am calling them as inhibitors, are shown to be inhibitors of multiple targets of SARS-CoV-2. For example, the remdesivir is already known because it is a target of the RNA-dependent RNA polymerase of the SARS-CoV-2. And in addition, we are saying that it could also target the main protease. And so your drug, there is a saying in many Indian languages, 2 mangoes in a single stone. So that's very attractive. So you can see, the lines, these crisscross lines, some of those attractive drugs are attractive because they are targeting the multiple critical proteins of SARS-CoV-2. And also there are many biological processes to do with host that are closely connected with the invasion of the virus. So for example, the blood coagulation is one of those factors strongly connected to the pathogenesis caused by SARS-CoV-2. And some of these molecules, for example, one shown in this purple here is also anticoagulant, known to be anticoagulant. And some of those drugs are given for the inflammatory response. So again, the human processes are highly connected to the viral pathogenesis. So it's nice to see certain molecules that we have proposed can act both on the human proteins positively as well as on the viral protease with a view to abrogate it. I think I will conclude my talk by saying that we suggest combination of drugs to try against COVID-19. As I showed in the previous slide, there are many. Some of the proposed drugs have multiple targets, or they also operate positively on the human response system, like anticoagulants. As any drug repurposing approach is based on the principles of polypharmacology, the effects of predicted drug on the undesired target need to be monitored with utmost importance. Our protein-centric protocol suggests some of the possible host off targets for the molecules that can target SARS-CoV-2 main protease. These findings provide clues in the designing of toxicity profiling studies. I almost forgot to tell that my student also explored the cross-reactivity with the human proteins. And one of the filtering aspects in our studies is, again, not to weigh too much on those inhibitors of main protease, which could also inhibit some human protein. After all, everything is computational, so it has to be pretty much followed up by the laboratory studies. Of course, our work provides the potential off targets in the human. Overall, our prediction shows some promising results which will contribute towards meeting the global challenge of COVID-19 pandemic and urges for laboratory validation. So all the work was done by the student shown in the left side, Shuhini Chakrabarti, and she really initiated all that in my group. And she's a very good expert. She comes from two backgrounds. She has done a B Pharm and M Pharm, followed by some extensive work on informatics, worked in companies and things before joining as a PhD student in my group. And Sneha Bhimireddy, shown in the right side, was ably supporting Shuhini towards the later half of our project. The paper is just out in the web on the Molecular Omics, the Royal Society of Chemistry journal. And my group is generally supported by Department of Biotechnology in terms of IISc DPT partnership, and thanks to DST in the form of mathematical biology and JC Bose fellowships to me. And I'm very, very open to collaborations. In fact, I would like to say that my good friend and colleague, Professor Ravi Sundaresan of Microbiology and Cell Biology Department, has very kindly agreed to test out some of the compounds, promising compounds that we proposed from our work, computational work, both on the cell lines as well as on the mice models, but we have to get lots of permissions and go through many bureaucratic steps. But there are a large number of experiments that we would like to actually to do. I'm extremely open to collaborations, both with academia and with industry. On this note, I would like to thank BIT again and stop my seminar and take the questions if you have any.
Yeah, thank you, Professor Srinivasan. Now this talk is open for questionnaire session.
Yeah.
Professor Vijay.
Yeah. I have a little bit naive question, very naive. Yeah. Is there any common protein, I mean, not exactly for total sequence, but some kind of a protein similarity between some of the tuberculosis and the COVID, even the already known structures?
Okay, I'm sure there are, because what we are targeting is cysteine proteinase with serine proteinase 4.
Yeah, that's the reason why I'm asking, because one of the protease inhibitor was already tested by one small company, Natco, and she has come out with some interesting data on this separated antigen from tuberculosis, which was simply done by precipitation. So I think you must be aware of that work. So I think, is that similar here?
I have simply documented as antibacterial in my tables and in my paper, a long supplementary table. I have not classified as which antibacterial that is. I'm sure there are some anti-MTB in our list as the potential target. Drugs against them.
Yeah, because she has designed a protease inhibitor for that. Yeah. And then I'm very much interested in what you are showing, whether these type of inhibitors are common to both. Yeah, thank you.
we can sit down together. I can bring out the details of my data and we could discuss one.
I think probably we'll come and meet you. I'll tell, you know, probably Supriya Kasikar. So I will bring her to you and then we will discuss. Yeah.
I don't know when Corona will allow us to meet.
Soon. Very optimistic. Very soon. Yeah. Yeah.
So here there is a question from one of the attendees. Right. The attendee is asking, Indian herbal drug Withania somniferous active compound is thought to inhibit SARS-CoV-2 main protease. Okay. Any comment on that? How difficult will it be explored to explore herbal drugs for antiviral applications?
Actually, I have not covered it at all in my seminar here, unfortunately, in the interest of time. There are quite a few herbal compounds that I have listed in our paper. I don't have it right now to show it to you, but there are herbal compounds. I mean, the compounds from the plant origin that are in our list as well, and they show high similarity to already approved. I mean, certain drugs already approved by FDA or under investigation. Yeah, there are certain compounds. We didn't particularly target the herbal drugs. And in fact, if you look into the paper by Professor Saudamani of NCBS, she has targeted, specifically the compounds from the herbal origin. So that paper focuses on. We do have some entries in our list.
Right. And another question is, how about trying these small compounds on binding to RNA of virus itself? Calculations, computational calculations.
Yes, this is only a beginning. We are extremely excited about our the non-planned entry into this area. But having got into it, why not go full-fledged? Sneha Bhimi Reddy is currently investigating a number of other targets, including non-protein targets in the SARS-CoV-2.
Right. So I don't see any more questions here. So if there are no more questions, I would like to thank Professor M. Srinivasan. And Professor Krishnan, do you want to ask any question? Yeah.
No, I just, so 2 or 3 comments. First of all, I apologize for stating that you received a grant. I'm sorry about that. I got it mixed up. It should be on the records, so it should not be, it should be correct. Whatever you said, it was correct. Okay. So secondly, I also would like to thank you for hosting Dr. Sabarish in your lab under INSA fellowship. We'll continue to collaborate. And one interesting comment by one of the students, she says, she says your narration about 36 Chambers movie, it was very interesting. It refreshed her UG lessons about Flipper Baby also. Okay, so she was very happy to hear from you and she could narrate it to your talk. Thank you so much. Thanks for your insightful talk. And I'm sure many participants will be excited to do this kind of work and then come out with new ideas. That's the purpose of this seminars. Thank you so much, Professor Srinivasan.
Thank you, Professor Krishnan. And I'm very happy to interact with students in particular. If my talk is, creating some spark in students, that's the main purpose of my talk here.
Sure.
I mean, the publications will come and go, grants will come and go.
Exactly.
It is more important for me if something works out, that is much, much better than any publication or any grant. If any of those compounds works and if I'm inspiring a student, and that's what will make me most happy.
Exactly.
And then my thanks to the student concerned for his or her admiration. Thank you.
Right. I need to put on the records that when 2 days ago, there were 900 registrations. Today morning, we opened our Google Drive, then we found 1,500 registrations.
Oh, okay.
Yeah, so it is live webcasted on Google also, YouTube. So, there are nearly 300 to 400 participants in the Zoom itself. So rest of them are coming through online in YouTube. Thank you so much. And it's a very exciting.
Krishnan, Dr. Srinivasan has always been a student lover.
Right.
Because I've been to his lab long time ago, full of students, everybody interacting. So that's so I learned a few things from that culture
Sure.
very early. Yeah.
Thank you so much. Thank you.
I myself is still a student. When I move with younger students, I feel young.
Exactly. That's what I also observed in you, and I like that. Yeah.
Okay. Thank you. Now we'll move on to the next speaker. Give me a minute. I'll just open the slide and then introduce the speaker. So Dr. Rishi Jain is the current Vice President Medical Affairs, Clinical Research and Regulatory Affairs from Wockhardt. And so thank you so much, Dr. Rishi Jain. So he's been serving in this company for more than 5 years. He's held key leadership roles at various companies and prestigious institutes. He's a medical doctor from Madurai Medical College, and of course, he has also done MD from All India Institute of Medical Sciences, New Delhi. He has several published papers, international journals, especially in the medical sciences field. He has won several awards and fellowships. Importantly, Business Championship Award, individual July 2016, M.U.R Pharmaceuticals, UXL Award in 2014 by Glenmark Pharmaceuticals, Certificate of Appreciation by Glenmark Pharmaceuticals, Telma Award again by Glenmark Pharmaceuticals, Certificate of Appreciation by Glenmark Pharmaceuticals. His research interest is in medico, marketing, clinical operations, pharmacovigilance, regulatory affairs, quality control, and of course epidemiological studies. Today he's going to share with us about his experience in artificial intelligence, how it could be adapted in drug designing and development. It's going to a new era in this artificial intelligence, which has been introduced into drug designing. We are looking forward for your talk, Dr. Rishi Jain. Thank you so much.
Thank you, Dr. Krishnan, for this kind introduction. And I would like to thank the organizing committee for inviting me today for giving this lecture. I will especially like to thank Dr. Sanjeev, whom I have personally worked with at All India Institute, for giving me this opportunity. I think that there is no right time then in the current world of pandemic that we resort to artificial intelligence and digital technologies to expedite the process of drug discovery. I'll just share my slides. I hope I'm audible to everyone, right?
Yes.
So artificial intelligence is a very upcoming area, and of course it has a huge significance, because if we look from a historical perspective, drug development has been a very long process. Coming from a pharmaceutical industry now, and I have seen all the three worlds. One is as a clinical practitioner, I'm a medical doctor. Then I had my stint at All India Institute in Department of Biophysics. So I have seen the lab works and the aspects of how do we get into drug development and various other technologies. And now I'm working with the pharmaceutical organization Wockhardt. And there we have a complete end-to-end capabilities of identifying a drug, doing the complete development of the drug, and then be able to manufacture it and commercialize it as well. And I'm very proud to say that very recently, Wockhardt Pharmaceutical has launched a new novel antibiotic, which is for gram-positive organism like methicillin-resistant Staph aureus. And we are the first organization in India who has a novel chemical antibiotic, which is available and launched now. And to re-emphasize on how important this is, 92 years back, penicillin was identified as the first antibiotic for bacterial infections. And as a country, we have taken almost 92 years to launch a drug from our own house. So this makes Wockhardt the only organization in India which has a product which is launched from Indian pharmaceutical company and which will be globally available now. So before I get into artificial intelligence and drug development, what I thought is, let me also give a quick snapshot of what we do at Wockhardt and how we are possessive and how we are fully into drug development and what are the capabilities we have and how does this converts into clinical drugs being launched in India as well as around the world. So right now, we already have 6 new chemical entities which are at very advanced stages of development. And out of this, 2 have been recently launched in India. One is an oral antibiotic and one is an injectable antibiotic. Very recently, we have launched. And of course, that comes with a long work of around 20 years. We have our own research facilities in Aurangabad, India, in Wrexham, UK, and Chicago, US. So this product was based on the work which we have done in Aurangabad for almost 20 years. We have a huge team of 850 scientists globally, out of which 150 scientists are based out of Aurangabad. And this is headed by one of our microbiologists, Dr. Mahesh Patel, who leads the research facility there. We also have capabilities of biotechnology development, and we were the first organization in India to develop recombinant human insulins and recombinant human insulin analogs like glargine. We have our own manufacturing facilities for erythropoietin, which is again a recombinant product. We have multiple abbreviated new drug applications at US FDA and European Union, and lots of patents have been filed. We have capabilities of end-to-end, which includes our microbiology, toxicology, pharmacology, medicinal chemistry, process scaling, analysis, formulation, new drug development systems, and including gene cloning, bioinformatics, protein modeling. So all these capabilities are there in-house, and end-to-end solutions are being provided in-house, from identification of the molecule to the development of the product and to the commercialization of this product. So it's a long journey. It's a vision of our chairman, Dr. Habil Khorakiwala, who is very passionate about drug development and research. And that is based on his vision. We have been working in this journey for almost more than 20 years. And now we have reached to a state where our 5 chemical entities are at very advanced stages of development. Another 2 have been launched. And all of them have been granted a coveted QIDP status by United States FDA. QIDP status is qualified infectious disease products, which makes the drug eligible for a fast track approval and also gets an exclusivity for additional 5 years after the patent expires. So these are our capabilities in terms of discovery. We have our own toxicology team, safety pharmacology, drug metabolism, medicinal chemistry and intellectual properties, antibacterial in vitro and vivo screenings, elucidation of new chemical entities, mechanism of actions and mechanism of resistance, and non-clinical pharmacokinetics. Coming to our capabilities in terms of antibiotic development, we have in-house clinical microbiology, NC product manufacturing, analytical technologies. We have full-fledged clinical team, which I heads. We have a regulatory team, which I heads there, NC scale-up and formulation development. This is just a snapshot of what global organizations are doing. Right now, if you see, there are around 16 products which are under various stages of development. And out of these 16 products, 5 are owned by Wockhardt, and other organizations like Pfizer, Merck, Roche, GSK, Novartis, all have one product in the field of antibiotic development. So Wockhardt is actually a global leader in terms of antibiotic development, and we are having the maximum number of drugs in the pipeline. And most of them are in final stages of development. And we expect that in next 3 to 5 years, all these products will be launched across the globe, including US, Europe, and India. Multiple patents have been granted. We work in a collaborative fashion with various international universities. So this is a list of universities. There are multiple universities under whose collaboration we develop these products. All the products have been recognized by WHO in their report on global antibacterial pipeline, and all our Wockhardt products are featuring in WHO report as the new chemical entities. This is a list of the Wockhardt research products: 7712349, 4873, 5222, 4282, and 6777. 6777 is the latest entry into this coveted list. Sorry, am I not audible?
Sir, you are audible, sir. You are audible.
Okay, thank you. So 7712349, the last one in the row, has been recently launched. It's available in India now, and the studies are ongoing for approval in various other countries. The remaining products, 5222, which is one of the best products for gram-negative organism. It's a destination therapy, and it has shown to be effective in all the multi-drug resistant gram-negative Acinetobacters and Pseudomonas. And this phase 3 is global phase 3 is going on. For 4282, it's again oral antibiotic and IV as well, and it will be used for hospital-acquired bacterial pneumonia and ventilator-associated pneumonias. And then 4873 is respiratory pathogens. So all the three are in various stages of development. And within the next 3 to 5 years, we expect that all of these products will be in the market. So this was just a brief snapshot of what work we are doing at Wockhardt in area of drug development. Although the topic today is about artificial intelligence, and overall globally, I will say everyone is in a very nascent stage of adopting artificial intelligence in the drug development. But then that's the future, where the future lies, and I think all of us should be cognizant of that fact that the future artificial intelligence is going to play a major role in drug development, in reducing the timelines, in optimizing our resources, and hitting the products very soon in the market. So we need to understand, adapt, and learn. It's a learning process for everyone, but I think it's very, very important right now that we realize that yes, this is going to be the future of drug development, and all of us need to learn more and more about it. So this is, I believe, one of the hottest tech trends. The healthcare industry is obviously often at the forefront of innovation and technological advances. And we have been at the healthcare industry always been eager to innovate, to find out new solutions to the therapy gaps which are available. Artificial intelligence has the potential to transform the diagnosis. That is again very, very important. Diagnosing a disease is very, very critical. And artificial intelligence has started playing a role already in the diagnosis of the disease. They are able to scan various pictures, and through those pictures, a diagnosis can be done by a machine. Treatment of the disease, they are able to personalize the treatment approach to the various diseases, and helping to ensure that patients get the right treatment at the right time. So artificial intelligence has already started playing an important role in all aspects of disease management, whether it is a diagnosis, whether it is a treatment, or whether it is the complete management of the disease or the patient. The market is exploding. Within the last 6 to 7 years, we have seen the market has grown by almost 10 to 11 times. So this is one of the fastest growing market, and that's what brings interest of all the commercial organizations, all the pharmaceutical industries, all the researchers, that they are looking a very, very positive approach in terms of how health artificial intelligence market is going to grow and going to give lots of solutions for us for the future. 106 companies, more than 106 companies have already started entering into artificial intelligence. They are working in all areas of artificial intelligence, whether it is patient data and risk analytics, whether it is research, medical imaging and diagnostics, mental health, drug discovery, virtual assistants, wearables, miscellaneous topics, inpatient care and hospital management, emergency room and surgeries, lifestyle management and monitoring. So I think this is almost the complete cascade of things which a patient and a doctor deals with. So everything is getting captured by interventions from organizations which are entering into artificial intelligence in transforming the healthcare. This is going to help us in surely reducing the drug discovery times, provide virtual assistance to patients, and diagnose ailments by processing medical images, among other things. The basic question is, what is artificial intelligence? It is nothing but the simulation of human intelligence processed by computers. So it is a predefined formulas and predefined analytics which is being fed into the computer system, and they are able to analyze the mammoth data, vast data which is available, and try to come with an analytics aspect that how this data correlates and how this data is helpful, or what are the trends we can see in these data. AI is a set of different technologies that mimic the cognitive functions of human beings. So the thinking process, which we always thought is a unique function of human beings, now what we are trying is we are asking these machines to mimic or to represent the cognitive functions of human beings and to analyze and interpret this information. The process includes acquiring the information, developing rules for using the information, drawing approximate or definitive conclusions, and self-correction. So this is a process where you first is acquire the information, you get the information, then you identify the rules. How do we use this information? But unless we have set of rules to understand how this information is usable, we will not be able to get or make sense of this information. And then how do we reach to a conclusion or make some approximate conclusions about what this information is telling about, and then self-learning and self-correction to improve going forward. So industry landscape, if you see artificial intelligence in healthcare, it has gone in all the aspects, whether it is patients aspects, whether it is doctor aspects, whether it is the research aspect, whether it is telehealth. Telehealth includes your telemedicine, lifestyle management, disease management. And the biggest surge what we have seen in the recent 3 to 4 months is the surge in the telemedicine. Telemedicine is a field where the patients will be able to consult a doctor digitally using virtual platforms. Although this was available in India since a long time, but the usage and the acceptance was at a very nascent stage. And after the COVID, the acceptance of telemedicine as a solution has significantly increased, and lots of doctors and patients are ready to consult doctors through a telemedicine platform. So telemedicine also has an inbuilt artificial intelligence tools which help the doctors as well as the patients in multiple aspects. We have chatbots. Everyone knows that now wherever we go, instead of a customer service representative, now everything first we deal with is a chat box. Again, these are all artificial intelligence tool which has a pre-fed information, and if you ask that information, there is no wastage of human resources, and that pre-fed information can be provided by these machines on various topics. Lots of wearables and devices for health purposes we are using. We are personalizing the genetics, like the genetic makeups can be evaluated and studied in aspects of mental health, women health, skin, and drug discovery. Drug discovery, information, clinical trials, genetic research, everywhere health, artificial intelligence is going to play an important role going forward. And coming to the doctors facing medical records, again, a very, very important aspect. How do we keep these medical records in place? Data analytics, medical imaging, and hospitals. So everyone is using tools which are artificial intelligence tools, and which are helping all the healthcare providers and the patients and the people who are involved in the healthcare setups, and able to assess these information and to analyze this information and to help patients in the best possible way. What makes artificial intelligence so suitable for use in medical research? We have so much data. Now it's like an information overload. there is a wealth of data which is available. And good part is now it is all in the digital format. Days are gone when we used to do clinical studies and patient data was in papers. Now everything is done digitally, everything technology is so advanced, and every data is available in a digital format. Just imagine if this COVID pandemic would have happened almost 30, 40 years back. And I think it would have disrupted the whole world badly. But still today we are seeing, although it has disrupted, but still 70 to 80% of things are working on an technology platform. So we are able to do this conference today because of technology. I am doing my complete work from home. Actually, I'm working more than what I used to work in office. And more organizations are understanding this importance of working from home using the technology. And giants like Novartis has made a globally work from home policy. All its employees across the globe can work from home. And that is not a temporary solution for Corona. It is a completely change in the way we deal with employees. They have said it is lifelong work from home unless you want to come to office for some other reasons, you can come, but it is completely work from home. So the way the technology has helped us is significant. It is huge, and in future the technology is the main key driver of whatever research we are going to do. It's going to help us in all the aspects, and that's why it becomes significantly important that we again change our tracks and adapt and accept that yes, this is the way to go forward, and try to learn and try to make best use of the available information and technology. So coming to that, we have enough of wealth of data available. Now it is the point is, how do we analyze this data? How do we make sense of this data? So obviously, humanly, this is a mammoth task. It will take years and years to analyze this data. And that's what machine learning comes into play. There are multiple advances in machine learning and analytical algorithms, and they are really helpful in terms of analyzing this information. Artificial intelligence has the ability to collect this data, analyze this data, and make sense of vast amount of unstructured and variable data. I think that is the most important aspect, to make sense of this information. Information is good, but unless you are able to get some conclusion, some results, some understanding that how does it help me going forward, or is it just a piece of information hanging, or there is something which I can correlate to this information, and can I use this information for my drug development, for my patient welfare, for my management of the complete disease spectrum. So that is the most important aspect. How do we make sense of the vast amount of unstructured and variable data, and quickly and often more accurately than a human being? And quite possible, I was seeing a video from Google, and what they have said is they can do retinopathy diagnosis much quicker, much faster, and much more accurately than what doctors can do. And this is correct. So obviously there is a subject of human error which is always possible, and with machine learning, when you have to interpret a large information, a large number of variables are possible, I think machine learning is a way which can help us in minimizing those errors. I just want to take you through how does the drug discovery process happens. I know you all are working in that area, but I'm not sure how many of you have a clear understanding of how the lab work gets converted into a commercial product. How are we able to launch a product, and what does it take as a pharmaceutical organization to launch a product, a new chemical entity or a new biological entity, to launch a new product in the world? If you look at the timelines perspective, the timelines can be anywhere between 15 to 20 years from the identification of the product. When you want that, yes, I will launch a product in this area, for example, a diabetes management or an antibiotic, rough around of time which is required to launch a product in market is 15 to 20 years. So you understand, I'm talking about a huge duration, which is really very, very significant. Coming to money, how much money do I need to spend? The money spent will be to the tune of $2 billion, and sometimes it could be even more. If I want to convert this money into Indian rupees, it will be around 15,000 crores of Indian rupees, which you will spend in development of a drug. So this is a huge money which I am talking about. 15,000 crores organization spends in developing a new drug. They spend almost 15 to 20 years. Patiently, they have to hear that, yes, my drug will come after 15 to 20 years. This is a huge amount of money. And then what is the risk in investing that money? 97% of the drug programs fail. So if I try to identify 100 drugs, out of that 97 will fail, and only 3 will be the drugs which I will be able to launch after a period of 20 years. So this is a huge risk which an organization has to take. And it is really difficult. Less than 40% of the known disease are currently treatable. We don't have treatment options for more than 60% of the disease. We somehow try to manage them. But can we cure them? Can we do complete treatment? Answer is no. We are just trying to manage the diseases, but complete cures and solutions are still at bay, and it's going to be a long journey when we are able to achieve that, yes, all the diseases are curable or treatable. New discoveries are extremely rare, mainly through academia and government research. So new discoveries are rare, and what we are doing is most of the organizations, they are playing with the known molecules. They are trying to change the structure, trying to improve the sensitivity, specificity of the molecules, trying to reduce the side effect of the existing drug, and see how we can repurpose these drugs. And the COVID is another example of repurposing. Most of the drugs which have been recently approved, whether it is remdesivir or favipiravir or itolizumab, all of them are repurposed drugs. They were already available and they were already used for multiple other purposes, whether it is hydroxychloroquine or ivermectin. We all don't know how effective they are actually, but they have been all repurposed drugs from existing drug therapies. And that's why repurpose becomes another important aspect of overall disease management. And that artificial intelligence can again play a very, very critical role in analyzing the available information of that drug which is already available, and then by machine learning tools, by our algorithms, try to understand whether this drug, based on the known mechanism, based on the known properties, can this drug be helpful in various other indications as well or not. So to summarize, what I just wanted to say is drug discovery is a very, very lengthy process, very, very expensive process, and sometimes very, very frustrating, and sometimes very, very discouraging and demotivating, because you need to spend a lot amount of time and money, and then the results are not sure, and not more than 3% of the patients, or sorry, 3% of the drugs are able to achieve the end outcome of commercialization. And many drugs, you know the history, many drugs, even after commercialization, has to be withdrawn from the market because of some side effects which we get to know only after the drugs become available or when it is used at a large subsets of patients. So again, it's a very, very lengthy, long trodden process. And again, it takes a lot of time and effort. This is just a snapshot. Sorry. So if you start with around 10,000 molecules, if you start around with 5,000 to 10,000 molecules, and out of this, after 3 years, you will realize that around 250 compounds of the 10,000 can get into are eligible to go into next stages. And you can think about doing some preclinical work. Out of that, after end of 5 years, you will realize that only these 5 compounds out of the 10,000 or what you started 5 years back are eligible to go into clinical aspects, which is your phase 1, phase 2, and phase 3 clinical trials. And then you start doing these studies, which takes you around 7 to 8 years. And at the end of 8 to 10 years after that, out of that, only one product gets an US FDA approval. So that is the only effective product which you were able to identify out of those 10,000 products which you started with over a period of 15 years and spending of around 1.5 to 2 billion US dollars. And where does this money goes? It's not only the drug discovery takes around 30% of the money, but the major amount of money goes into the clinical trials. And phase 3 is the one where maximum amount of money is being spent. Phase 3 is that phase of the clinical trial when you want to test that the drug is safe as well as effective in human population. So you do a study of the actual patients and you expose the patients to this drug. And then you take it with a comparator arm, whether it is a placebo or a known established therapy against that disease. and against that you compare your drug, and then at the end of the phase 3, you are able to establish whether my drug is safe and effective for the management of this disease. So it's a long process, takes a lot of time, lots of money, and then we are able to get a product approved. About half of this investment is spent on clinical trials, with phase 3 trial being the most expensive and most complex. What is the success rate for clinical trials? 40% of the compounds advance from phase 1 to phase 3. So if there are 100 compounds which go into phase 1, out of that only 40 are able to enter into the phase 3. So 60 get out at this level, and you realize by this time already a 50% of the money has been spent. After spending 50% of the money, you realize that this product cannot go forward. 1/3 of the compounds advance from phase 2 to phase 3. So out of 100, around 30% of the compounds will enter phase 3. And only 2/3 of the compounds advance from phase 3 to FDA approval. So from phase 3 to FDA approval, again, only 2/3 are able to get in. So if you see overall, around 10% of the compounds entering the clinical trial stage advance to FDA approval. So if at this stage, when you are entering the clinical phase 1, 2, 3, if there are 100 products which enter, out of that only 10 such products will get approval. So of course, there is a huge filtering happening. There is a large number of molecules which we have to reject because of the safety concerns or efficacy concerns or any such aspects. And that's why we realize that it's not a simple process to develop a drug or a vaccine. Almost similar aspect is in vaccines as well. People are trying globally hard to develop a vaccine for COVID-19, and it's been a long journey. People are trying to cut down on the timelines in all possible ways. Still, we see that it will going to take some time when we really have a solution to this. Artificial intelligence has the potential to transform the drug development process, making it more efficient and more effective, thus benefiting all the parties involved, from the companies developing new drugs to the patients in desperate need of viable treatment. So of course, artificial intelligence has the potential to transform the complete process. It can improve the efficiencies and effectiveness of all the parties, whether it's a pharmaceutical organization or whether it's a research scientist developing the products. If you want to compare your traditional approach versus artificial intelligence approach for drug designing, traditional approach is a target driven. You identify a target, you identify a receptor, and then you work on identifying the molecule or the structure of that receptor and identifying a drug which can block that receptor. So it's more of a target driven. Artificial intelligence is more of a data driven. You learn from the existing information, you learn from the existing data, and then you drive solutions out of that. Traditional work well for easily druggable target that have well-defined structure and whose interactions inside the cells are understood in details. So it only works well for targets which you can identify, which you can make structures, you can crystallize, and then you can understand their interactions, and then you can try to inhibit these targets. But artificial intelligence works with complex algorithms and machine learning and can extract meaningful information from a large data set. So it works in all kinds of scenarios, and it is helping in developing complex algorithms and machine learning and can extract meaningful information from a large data set. Traditionally, it is extremely limited due to complex nature of cellular interactions and limited knowledge of intricate cellular pathways. It is very, very difficult to understand what happens when a drug enters into the body system, what are the cellular pathways, how the drug gets metabolized. All these aspects are not very easy to understand. Artificial intelligence identify compounds that could bind to undruggable target as well. That is proteins whose structures are not defined. We don't know the protein structure. Again, identifying the protein structure is again a Herculean task, and I have myself dealt with this. It's not easy to identify a drug structure. It takes a lot of time and effort, but artificial intelligence can help us in even identifying compounds that could bind to undruggable targets, that is proteins whose structures are not defined. A predictive sets of compounds that can be easily identified with artificial intelligence in relatively small amounts of time and at a quarter of the cost of traditional medicine. It overall helped us in reducing the time, again, which is a most important aspect. And second most important aspect is the cost, the amount of money which we spent in traditional drug discovery, which we discussed was around $2 billion. It can almost reduce it to 1/4, which is a huge saving of 75%. And then it also helps in reducing the timelines. Artificial intelligence offering in drug discovery, it is faster, it is cheaper, and of course more efficient than the traditional drug discovery. Development of drugs that are more effective. Personalized healthcare, again, it is very, very important. And in today's world, everyone is talking about personalized healthcare. What is personalized healthcare? Personalized healthcare is one size does not fit all. Every patient needs to be treated as an individual patient, and the treatment approach, the management approach has to be customized, has to be individualized for that person. And that's what you mean by personalized healthcare. Your traditional drug discovery only targets as a complete class, as a complete group, but it doesn't give you an opportunity to personalizing your treatment approaches. And artificial intelligence is going to help us in a long way in terms of personalizing the healthcare. Easy access to effective drugs and treatments at a much lower cost. And of course, it will improve the access to these drugs and also reduce the cost at which we are going to develop these molecules. How artificial intelligence will assist in drug discovery? It will help in finding the new insights into a disease. So this is the basic thing. Unless you have a new insights or you don't understand how the disease behaves, what are the requirements to manage this disease, you are not able to identify a drug target. So you have to get new insights into the disease. It helps in identifying and testing drug compounds to determine their possible beneficial effects and repurposing existing drugs against different diseases. So all these aspects, artificial intelligence can play an important role, and it will make all these processes easier and faster. What are the benefits of applying artificial intelligence to drug discovery? Subjective bias and existing knowledge is not a factor. So again, your knowledge is not important. What is important is the amount of data what we have. There is no bias. This is all machine learning, and they are going to do as per the algorithm. So there is no individual or human or subjective bias. Knowledge is not a factor. What is important is the information what you have already available. Artificial intelligence utilizes the latest advances in biology and computing to develop state-of-the-art algorithms for drug discovery, reduce potential for false positives, and shortlist promising targets without the need for extensive experimental input and manpower hours. So it reduces your manpower hours and it reduces your extensive experimental inputs and can help in making these drugs available much faster. The applications of artificial intelligence and its subfields like machine learning and deep learning in healthcare. So artificial intelligence again is a very broad topic. If you further see, there are deeper aspects like machine learning and the deep learning, and which I'm obviously not going to go in detail because that's just to give you a snapshot of how artificial intelligence can be helpful. If you look at this side of the picture, artificial intelligence has a significant role in healthcare, in research, wearables, drug discovery, virtual assistant, patient data and risk analysis, hospital management, mental health, surgery, lifestyle, and digital health monitoring and diagnostics. And that's what is helping us in our day-to-day patient care these days. Artificial intelligence can be applied at various stages of drug development, starting from identification and validation of the targets, designing of new drugs, drug repurposing, improving the efficiency of our research and development systems, and aggregating and analyzing biomedicine information, and refining the decision-making process to recruit patients for clinical trials. So on all these aspects, artificial intelligence can play a very, very important and significant role. Just to summarize artificial intelligence in drug development process, it has essentially role in the complete drug development process, whether it is early target validation, biological assays, compound screening, hit lead identification, lead optimization. So this all is part of your target-based drug discovery, and then preclinical development, phase 1, phase 2, phase 3, and phase 4 studies, opportunity for serendipitous identification of new therapeutic uses. So in all these aspects, throughout the drug development process, artificial intelligence can really help us in various aspects, like benchmarking compound site design, predict target role in a disease, design of all in silico compound libraries, identify novel targets, predict druggability of targets, and predict signaling systems, prediction of structure activity relationship, what we call SAR, and prediction of ADMET properties. What are the properties of the drug in terms of absorption, digestion, metabolism, excretion, and things like that. Drug repurposing, selection of patient population in clinical trial to increase success rates. And pharmacovigilance, the study to evaluate the drug safety and the side effect profile. All said and done, although artificial intelligence is giving a lot of hope and it's going to be the way how drug development will happen, but right now, even in the early 2020, this is at a nascent stage, and we expect in next couple of years, we are going to talk more and more about AI in all the aspects. So the future is in AI, going to take some time, but definitely it has already started making inroads in all aspects, whether it is pharma industry, whether it is drug discovery, whether it is regulatory aspects, clinical operations, pharmacovigilance, as well as supply chain. At Wockhardt, already in supply chain, pharmacovigilance, clinical operations, we have already started using artificial intelligence at various aspects. And then we expect and we are very hopeful that this is the way it's going to help us in future. Going to reduce human efforts, going to reduce human time, and of course, it's going to optimize the cost which the organization is incurring in all these aspects. This is a survey findings which also tells about why artificial intelligence has not been used by a major population. So the most important reason which came out was the lack of skilled staff. People are not well trained. They are not aware about using technology, using artificial intelligence as a tool, that how it's going to help. And that came out as a first and the foremost most important reason. Second was the safety, regulatory, and compliance concerns, followed by budget constraints, mistrust of unvalidated technologies, company culture, and legal and intellectual property concerns. So these were few of the reasons why people were not able to use artificial intelligence, but I am very sure very soon people are going to adapt this full-fledged. What are the artificial intelligence staffing predictions? Most responders, 59%, reported that their organization was planning to increase staff for artificial intelligence use or implementation within the next 1 to 2 years. So most of the major organizations are going to increase the staff who are well-versed, who are well-trained in handling AI tools. And that's what the trend will be in the next 5 years. If you see various giant pharma companies, global pharma companies like Allergan, Estelas, Bayer, Biogen, Roche, GSK, Teva, J&J, Janssen, Pfizer, Novartis, AstraZeneca, Takeda, Merck, Sanofi, AbbVie, everyone, all of them have already partnered with various AI-based organizations because they have understood the importance of AI in expediting the drug development, and everyone is now banking on to take advantage of this. Many of these companies already have invested in these tech companies of AI and have already joint ventures with artificial intelligence companies. Just to give one example, this is a Hong Kong-based startup, In Silico Medicine, using system called generative transorial reinforcement learning, was able to find 6 treatment targets in just 21 days using artificial intelligence, and one of which showed promising results in experiment involving mice. So you see the 5 years time which our normal drug process was taking 5 years to identify a molecule which can at least go into animal study or animal models. They were able to do that part in just 21 days. They were able to identify 6 targets. So definitely there is a big future for artificial intelligence in drug discovery. And very soon people will learn and understand this, and they are going to utilize this in their day-to-day working. So just to summarize my talk, this is my last slide. Artificial intelligence powered healthcare are driving meaningful changes across the entire patient journey. So it is not only about the drug development, it is about the entire patient journey, from diagnosis of the disease, from the management and treatment options available. Machine learning and artificial intelligence aren't concepts of future anymore, and they are getting into each and everyone's life. They're getting into the drug development, they are getting into the pharmaceutical organizations. Paradigm shift in the way patients today are treated by medical practitioners across the world. We are seeing a huge shift in the way patients are treated and managed. Artificial intelligence has the potential to transform medicine. Currently, there are no developed drugs that have utilized AI approach, but the future is very promising, and we expect very soon we will first see a molecule which has been developed using artificial intelligence. For an individual to be efficient in drug development using artificial intelligence, the individual should know how to train algorithms requiring domain expertise. So the training is very, very important. We need to understand and learn how to get best results out of artificial intelligence tools. Despite the benefits of AI in speeding up drug development, real experiments will always still need to be conducted. Definitely, we will have to do human clinical trials to understand what the predicted drug by artificial intelligence can actually be used in human beings or not. But definitely it helps to reduce the timelines. Definitely it saves on the money aspect and saves on the human effort aspects. With this, I would like to summarize and thank you by closing with a quote. Organization face 2 big challenges today. One is artificial intelligence and second is natural stupidity. With this, I would like to thank all of you for patient listening and inviting me again to share my opinion on this topic. Thank you.
Yeah, thank you, Dr. Rishi Jain, for a nice talk. Now the session is open for questions. Professor Vijay.
Yeah, I have to say, yeah, I mean, I'm little bit surprised with this natural stupidity. Of course, I've been with Wockhardt since long time. Sahib is a very good personal friend of mine.
That was on the lighter side, madam. That was a joke. It was a.
I know. That's why I wanted to know what is it COVID that you are mentioning, or which are the natural stupidities?
No, it is just a joke, ma'am. There is no meaning to that. It is just a joke that this is one of the challenge.
No, meaning is COVID.
No, it is not
the
COVID,
natural stupidity. Yeah. Okay. Yeah, I have one serious question now regarding artificial intelligence. See, this artificial intelligence basically is on the pattern, I mean, recognition of some pattern. So how would you decide if you don't have the previous data? That is what we are facing the challenge now.
So that pattern need to be identified. For example, if you want to identify a drug for a diabetes, and there are various.
That I know because I've been some of my friends are into this artificial intelligence, pattern recognition and other recognitions. But then the problem is if you do not know, unless and until you do that by experimental data, how can you foresee this type of pattern or that type of pattern? Or is it hypothetical pattern?
No, these are first to start with are a hypothetical pattern only.
Okay,
If you feed a large number of data and then the machine will come back that this is the pattern what we are seeing in your data. And then obviously a back correlation.
Okay, thank you. You have answered the question. Thank you. That is exactly what I wanted to know. Okay.
Yes, thank you.
Okay, there is one question from an attender. He wants to know, in artificial intelligence, how neural network can be used for predicting retrosynthesis routes and for diagnostics. Neural networks. Neural networks.
So, I don't know what does he really want to know. Neural network are human living system networks. So what artificial intelligence is going to do is you have to give him the information, whatever available information is there. Sorry, it's a machine learning process. So whatever information, even if you want to give information about the human CNS system or a neural network system, how does a receptors work, or how does we have lots of transmitters, neurotransmitters, on what all receptors they work. So all the information, whatever we have to feed it into the system, and then the machine learning or the artificial intelligence system can give us a trend or can give us a pattern that this is how it behaves. For example, when there is a excitatory neurotransmission, it obviously goes through neural receptors, and then they will tell you that when there is a neural transmission, this levels increases, this level decreases. So based on that pattern, we'll have to analyze, and we have to take that forward accordingly.
Right. Okay, and another question is how artificial intelligence helps to develop a drug from herbal and Unani single drugs or formulation during clinical practice, which already patients are taking.
Yeah, so again, if lots of patients are taking, there is a large number of data which becomes available, which we say as a phase 4 or a post-marketing data. And if we feed all that data into the machine, the drug, so sometimes what happens is you don't know that this drug is more effective in only disease X, or if along with disease X, patient also has disease Y, the drug may be more effective or less effective, or the side effect, if there are very, very rare side effects, you may not be able to, sometimes patient don't report, or sometimes you miss that. For example, in 1 lakh subjects, if a drug is given and there is only one side effect, you will not be able to pick up that. But because of machine learning, even those rare side effects or those rare correlation between two disease can be picked up with the amount of data. Because otherwise what happens is we are not able to analyze that large, vast amount of data and are not able to get clinical significance out of that information.
So which means that artificial intelligence can be used even for studying herbal and Unani drug formulations.
Yes, it is just a machine learning.
Coming to that question, sir, that is again a continuation of this question. See, I have proposed long time ago to see the correlation between the Ayurvedic thinking. Ayurvedic thinking in the sense the people are patterned like according to their vada gunas, okay, gunas and doshas. And can we really pick up those data and then try to find out what will be the uniqueness of one given particular drug, and then see if we can apply that in terms of recognition? Because I think that may be related to our DNA pattern. This gunas, doshas, and everything in Ayurvedic system. Can we apply that with your approach of artificial intelligence, that is machine learning?
Ma'am, I don't have much details about Ayurvedic information, but from a basic concept purpose, answer is yes. If we have the information available, machine learning has the capability and potential to analyze this data and give us the guidance in those directions.
Maybe this is high time that we should come together, because I've been proposing this, and one person, Dr. Govindarajan, I forgot his name completely. He has done that work partially. So I think this is high time people like you, Wockhardt, being a very big giant in the field, should get into this for using machine learning process to see how efficiently we can use it. I'm sure Ramesh Warrior is listening to me. He knows, and he was the one who was very much interested in that. Yeah.
Sure. We are already collaborating with multiple international organizations And if any such project is there, we'll be keen to explore the.
I think we should probably think of it. I'll come back to you with Ramesh Warrior and discuss.
Sure,
Yeah.
sure.
Dr. Naidu has a question, I think he's been. Please, sir. You need to unmute. Can you unmute, sir?
Dr. Naidu, can you please remove the mute symbol?
Still, we are not able to hear you, sir.
Is the mic working, sir? Mic.
We are not able to hear.
No, yeah, I think he is not unmuted.
Mic issues. I think he was unmuted, but I think some mic issues there.
Dr. Naidu may type the question.
Yeah, sure, sir. Sure, please.
Yeah.
Please. In the meanwhile, as he's typing the question, some of the students, they've asked similar kind of question, which is on undruggable target. So often, people encounter undruggable targets based on experimental evidences. Does AI help to pick up those things and then kind of a reductionist approach can we approach through AI for this?
Yes, definitely. That's what in one of my slide also I showed.
Right.
Undruggable target is the best advantage of AI. It can help us in that. Definitely, it can do that.
Dr. Rishi Jain, I have a question.
Yes, sir.
In diabetes, we all know that the insulin receptor phosphatase are the excellent target for diabetes, beyond doubt. But till today, no chemist in this world can make a compound you can make in the test tube picomolar level, no problem. But to work in the animal in vivo, to pass through the cell membranes, why can't artificial intelligence can do something about this?
Question is not very.
No, definitely. These are the questions which going forward, we will take help of artificial intelligence. That at what aspect this drug is failing. So obviously, I agree that receptors we know. We have identified the receptors. We are able to identify the drug. But if the drug in vivo system in body, if it is not working, then where is the drug getting either disintegrated, or why the drug is not able to give the clinical effect? And when we have all this information available, definitely we can take advantage of artificial intelligence to answer such questions.
Still.
Okay, so.
So Naidu's question.
Yeah.
No, he's asking, but I think it is still in mute only.
But he's chatting. He has typed a question in the chat window.
Let's see.
Can you call Krishna Naidu and tell him?
Right. Okay.
Yes, Krishna.
Him.
Sabrish.
Yeah, tell me. Yeah, Sanjit.
In between, you can take one question, maybe.
Yeah, okay.
That,
Yeah,
Dr.
fine.
Amit Alok has asked one question. You can ask one question.
Yeah.
You can take one question.
Yeah, see, the thing is, usually which kind of languages are used in artificial intelligence, computer languages? Like C, C++, or Perl, or Python?
I don't think I am the right person to answer this because it's more
He asked,
done by
he wants
IT
to know,
people.
he wants to know whether it's audible. Hello?
No, we can't still hear him.
Repeat the question, Krishnan. Ask him to get the question to you and repeat it.
Sir, you can just put his phone on speaker mode. That's all.
Yeah. So one student is asking, what should be
7771. Okay, I'll tell you.
Right. Okay.
Yes, section.
Right. One student is asking, what should be the prerequisites if to learn artificial intelligence as a MD pharmacology student?
Thank you.
Yeah, for an MD pharmacology, I think lots of basic books are available about artificial intelligence and how it is going to help. So obviously, this is not part of an curriculum in the medical colleges. So it has to be an additional effort which the student has to take in learning artificial intelligence. And a lot of information is available on internet. Lots of books are available. So it's more of a self-reading which they will have to do for this topic.
Right. Right.
Yeah.
Thanks.
So there's some technical issue with Dr. Naidu's this thing. So he told me to, he has a comment to tell. Apparently, he showed a stage 1 trial, human clinical trial. So the W771. Apparently, they have completed the clinical phase 1 trial in 2019. So they are ahead of it, and so this is the information he wanted to pass it on to you.
Yeah, so this is WCK771.
Yeah.
As he's saying, is one of our antibiotics which we have recently launched in India.
Right.
It is so we had two molecules, 771 and 2349. 771 is an injectable antibiotic, levonidifloxacin, and 2349 was an oral medication, oral tablet, and both are targeted towards methicillin-resistant methicillin-resistant Staph aureus. And very soon, very recently, last month only in May, we have launched both these drugs in India.
Okay. So, Dr. Naidu he's a director of Natco Pharma.
Okay.
Yeah, so this is the message he wanted to convey. Okay, so thank you so much for opening up an important topic, and it is going to help.
The future is there. Yeah,
futuristic medicine, it is very important, and then if it is going to cut the cost and time, this is the major bottleneck that we have in drug discovery development. I'm sure many students will be very excited to learn about this and then find new path for their building their career. Of course, pharma industry is setting a stage for these kind of new approaches, and then I'm sure academic institutes will follow the suit. Thank you so much, Dr. Rishi Jain. So we'll move on to our next speaker. So I'll just share the slide and then introduce Dr. Balram Patro. I hope you are all able to see the slide. Dr. Balram Patro has started.
Yeah, I'm here on. Yes.
Right. Dr. Balram Patro has started the entrepreneurial endeavor in 2014 and started GRK Research Laboratories and GRK Fine Chemicals Private Limited. So he's a CEO of GRK Labs in Hyderabad. Okay, so also he was senior vice president and head of chemistry services earlier at GVK Biosciences, and he served as a CEO and vice president of Chemgen Pharma Research International Private Limited, Chem Biotech Research Private Limited, Calcutta. He has done his PhD in chemistry from Northeastern Hill University, Shillong. He was a holding Humboldt prestigious fellowship at University of Constance, Germany. He worked on area of carbohydrates chemistry. He has more than 40 publications in peer-reviewed journals, and then he has written book chapters from academic press. Expertise in the area of medicinal chemistry, scale-up chemistry, analytical chemistry, areas related to material science. And then, he's worked with more than 90 international clients in the field of drug discovery research and large-scale synthesis. This is very important. He has participated in a core number in building the 2 largest discovery CROs in India. He started with a small group of 100 people and increased the team size to 900 people in GVK Biosciences. He has contributed significantly in different projects pertaining to synthesis chemistry and important drug intermediates. Over to Dr. Balram Patro for your talk. Thank you so much.
Thank you, Dr. Krishnan. And I would like to share my screen. I hope my screen is visible.
Yes, sir. Yes, sir. Please go ahead. Yeah.
So almost I am very close to the lunch. I think people have hungry of listening, also to hunger as well together. So I will be bringing some of the topic such interesting, so that we can balance both the phenomena. So pharmaceutical innovations and urgency to tackle COVID-19 situation. The recent, the world is facing a very different scenario. Actually, in most of the time, we had drugs available, we had the situation was there with us, and at the same time we had medicines. So the doctors at least had a weapon to fight the war. But unfortunately, in this pandemic situation now, we do not know the exact medicines. We don't know, we don't have vaccines, and we are in such a situation now. We need to tackle this situation and differently altogether to normal practices. So I would like to briefly talk about what is drug discovery. It's it's more like a blindfolded man taking out some of the molecules. That means, see, as I I just put in a cartoon, but it's not so easy. It's not blindfolded, but you need to know. I meant actually, you don't know exactly where is the innovation, which compound can be a drug, and how we can bring, and how can we bring a drug to the market. So I'll go through the little bit of the drug discovery process. So it starts from the target identification. Presently, suppose if you see, we don't know the target, although we know the AC inhibitor, also the spike protein and all, but we can't go directly to the virus and work out the binding part. So the target identification and compound screening. So these are the stepwise things happen. First, we do a biological testing, target expression and functional assays, and then we do this target ID, making the genomic and proteomics the proof of concepts are done. Then after that, the target identification is done. After that, we start compound screening. So this compound screening can be 2 part actually. Just before my talk, actually, there was an artificial intelligence discussion. It was ongoing. Actually, in drug discovery, what is artificial intelligence? One of the most important artificial intelligence is molecular modeling. That means computer-based drug design. That means, see, we take the target and logically we prove the compounds which are virtual, either through a small molecule or a large molecule. We can do the binding and we can see their scores and the drug-like properties, and we tell them that they are they are drug-like property, drug-like molecules. So after that, we can synthesize. The other way is also we can FDD, fraction-based, where actually we can take the small fractions and we can see the binding. After that, we stitch them together and we create a bigger molecule. So these are a couple of like computational chemistry, which are actually we can put into them into the artificial intelligence. And after that, there are also like preclinical testing of lead compounds. But in this compound screening stage, actually, we can do also the libraries, actually 5-6 million compound libraries. We screen them. We can do with computationally. Sorry, we can do direct. Those are physically, actually, those compounds are available in the library, and we can take out the binding of those molecules. And after, once we come through this, either through virtually or could be by real molecules, we can do a set of preclinical testing for the lead compounds, and which goes for the, actually that is proofs, that gives the proof of concept and discovery stage. And after that, we go to the phase 1 to 2 trials, and then phase 3 trials, then cells is the phase 4 trials. So this is the flow diagram of a drug discovery process. And next I go to the timeline and the number of compounds. Like, the initial, the drug ID, target ID and assay development, lead generation, it takes almost like 3 to 5 years. And we have to screen a couple of million compounds to come to around 10-15,000 compounds. And after that, we get some proof of concept. In the preclinical stage, actually, we come to around focus libraries, where actually we target different series of compounds, and we refine those series, the molecules which have showed some of the activity either in the our CADD approach, or could be by the assay approach, direct screening library assays. And those molecules we can go to the, it takes almost 1 to 2 years, and during this period we have to do the in vivo toxicity, and also ADME data, PK and PD data, all these things takes almost like another 1 or 2 years. After that, phase 1, phase 2, phase 3 molecule, it takes 6 to 7 years. So a new drug comes to the market for regulatory approval, and one new drug comes in, and that also takes 1 to 2 years during this regulatory approval. So basically, if you see, almost we have taken almost 11 to 14 years of research to come bring a drug. So we don't have that luxury of time, the drug, at this point of time, because we need a very urgently molecule which can be, which can work, which can slow, which can improve the health conditions of the, at least the people who are suffering in a later, in worst case scenarios. So almost we have to spend $1 billion to $2.5 billion per each molecule. So it's not easy time, and we don't have that time as well, that much time. So next, what we can plan is either the clinical candidates and all those kinetics, potency of the molecule, as well as lab and doses, all those things, it takes lot of time. So in this follow up, actually, at the present situation, we cannot take in a new drug. A new drug, making a new drug, almost this, I have given a drug discovery cycle, where actually the compound collection, primary assays, high throughput assays, and then chemical synthesis happens either through design or through the other way, as I said, computationally. And we come to the lead compounds, SARs and all. And all these process, which takes such a long time, and to bring in a clinical candidate. And after the clinical candidate comes out, we have to go for the clinical testing part. Clinical testing, again, it is almost similar time, almost 7, 8 years. So, and so that's why the total, if you see the drug discovery, drug development process is almost like, we have such a long time and as such a huge costing purposes. So that's why in this situation, like whatever we do, this medicinal chemistry or the computational chemistry, and then again development process. In the development, that is itself is a long part. Actually, we have to do the proof phase one in the healthy volunteers. After that, phase 2, phase 3, and all these again we have to see the toxicity part and side effects and the any other issues or some kind of other disease or side effects which creates some kind of big problems. So all this part actually it is done in the development side. So we have actually, like I have given one image of a virtual screening process where actually we have taken in a 22,000 library. We do the doc scoring and we did this the post analysis, and we found out these molecules. Out of these molecules, which are the best molecules, which are the lead-like properties. And after we took those lead-like properties, 10 virtual hits we have chosen. And after that, we have synthesized those molecules and we have gone to the biochemical assay. So during the assay, we have got almost like out of 10 molecules, 3 hits came below 20 micromolar. And then after that, we have gone to another 7 compounds and we have created new molecules as well. And all these molecules, then we identified and refined those molecules and gone to the a proof of concept. So this is normally the medicinal chemistry, and these are also quite expensive and time taking, and so that's why we have to identify these molecules. So it takes almost like 3 to 5 years minimum. And so in this present COVID situation now, what is we don't have the that much time. We don't have a time of 10 years to bring a drug, and now we don't have that much time as well as effort, all those things. So what is the best way is how do we can come to the drug repurposing? So that suppose in case already an existing drug, what is a drug repurposing? So in this actually we are taking the molecule which is novel already, which is already existing, which has, we know the it's some of the data toxicity data as well as phase 1, phase 2 data. And so normally in the drug repurposing, we have earlier as we have gone, I have updated that there are a lot of high attrition cost and all, but in this the situation is much better because already we have the molecules, we already profiled the molecules, we know the molecules how they behave, and they are all drugs, they are utilized for some other disease. So we have almost idea about their pharmaceutical profiles. So that's why that identification of this repurposal drug candidates, but there are also certain norms we have to follow them. We can't use all for anyone for anything. There are some of the examples, most importantly, either see the advantage of this is actually already because this is the existing drug, the availability is much easier. Second thing is we are sufficiently safe in preclinical models and in the human uses, and the time is very less because already it's an available molecule, and less investment is needed. Only some of the regulatory and the phase 3 clinical we have to do them, and substance like, that's hard. You might have listened to the most of the talks. Like, there are some molecules which show some efficacy, and some molecules, they do not show that much efficacy. And although they could be similar area utilized, but it has some of their binding or some of the way of like, acting is different. And the estimated, actually the cost is much lower compared to our $2 to $3 billion investment if you see in a drug area. But whereas in this one, it is almost like, more or less 10%, 10% only, 300 million is the investment required. And there are some of the examples which actually has previously utilized, like, for example, aspirin, which is an analgesic, but it is used for colorectal rectal cancer in 2015. Similarly, sildenafil, which is an engine, like, which is used for erectile dysfunction in 2000. And thalidomide, actually, it was used for the morning sickness, but in 2006, actually, it was utilized for the myeloma, like, lympho. It is a cancer, anti-cancer compound. Similarly, zidovudine, which was cancer, and it is in 1987, it has gone to HIV and AIDS. Similarly, like, minoxidil, which is a hypertension drug, but hair loss in 1988. Similar other molecule is celecoxib, which is pain and inflammation, and again it has gone to familial adenomatous polyps 2000. Similarly, like, there are other molecules like rituximab, which is various cancers, but it has gone to rheumatoid arthritis. Reloxifene, which is an osteoporosis drug, and which has gone to breast cancer in 2007. So during this, like, we already had some examples, some experiences. So now during this pandemic situation, and the mankind has taken a very quick steps, and we started a lot of molecules, drug repurposing. So some of the pipelines, development of pipeline indications, some of the hypotheses were generated in this area, and the mechanistic aspects also studied, and there are a lot of clinical trials came very quite faster, and there are some areas which actually showed us, like, urgency for treating COVID-19 is when we expect new treatment options. One is the repurposing drugs. We have actually a small like, set of molecules now has showed some of the activity, and now they are utilizing them. And similarly, antibodies, now the other area which is coming up is the plasma therapy, and like, they are creating the people who are surviving and creating the antibody, creating a defense mechanism inside their body. They, after they become healthy, they can contribute to the antibodies to the sick people again. So it's a body is a unique mechanism, and it produces the antibodies without any misery. So it's a way to supplement the testing, as well as inject the antibodies to the people who are in a very serious cases. And the other one is the vaccines. Vaccines is already going on. I just took some from the Bear Healthcare website. They think it takes a little longer time, like, the phase 1, phase 2, phase 3 in the vaccines. Although we see some of the fast development things are happening, and a lot of people are coming up with even the phase 2, phase 3 studies also has come up for some few of the vaccines. And so the urgency for treating COVID-19 is, one is repurposing the API molecules, which is much easier. And we work another area is actually drug repurposing, checking 3 in silico. That means what we are doing is we are taking in silico based, computational based, most of the molecules, we are taking them existing drugs and seeing their scores and how they behave, or if we can do small changes. But if we do some small changes, mean they become new therapy, so new drugs. So there is we need to go to the total activity again. So the other thing is the combination therapies. Combination therapies means some of them are antivirals, and some of them are like HIV drugs, or could be some of them are anti-cancer compounds. And with the antibiotics together, we are combination therapies we have created. And another is, as I said you, that the antibody research and the vaccine research. So these are the 4 or 5 areas we need to work, and we need to bring some changes. And I have put a couple of molecules which are gained some kind of repurposing in the drug environment, actually, like the azithromycin. Azithromycin is a macrolide antibiotic, and it is used as a combination therapy. And similarly, baricitinib, that is also a rheumatoid arthritis and JAK inhibitor. And this molecule, as well as then camostat. Camostat is actually a molecule from Japan, and this molecule is already a typsin inhibitor for treatment of chronic pancreatitis. And the other 2 molecules, which are chloroquine and hydroxychloroquine, which India got lot of name, but whatever we have done the testing in the molecular modeling part, we did not see much binding these molecules, but they are at least they were much poor binding score compared to some of the anti-HIV molecules. So the next is pobicistat, and that is also an anti-HIV and cytochrome P40 inhibitor. Similarly, darunavir. Darunavir is an antiviral and HIV protease inhibitor, and this is it's already existing HIV molecule. And darunavir. Then favipiravir, we have made this molecule as well, and this is already some of the pharma companies from India already they brought the drugs, and it shows some of the activities. And although there are a lot of debates on these molecules, some of them they are not 100%, they are not the medicines, but they are at least control the damage. Similarly, the fingolimod is an immunomodulator, and lopinavir and ritonavir, again these 2 molecules are antiviral and AIDS therapeutics. And lopinavir is actually, it is lopinavir, ritonavir, and azithromycin, or another large molecule, together they administrate. And remdesivir, again, it is a Gilead molecule, and it is an RNA polymerase inhibitor, and for Ebola and coronavirus. And during I think 1995, 2009, I guess, it has come forward, but ultimately there was some research has been done recently, and they are even permitted some of the Indian companies to make the remdesivir, and which is showing some of the efficacy in controlling the very serious cases. And ritonavir, again, it is an anti-HIV AIDS therapeutic antiviral, and this is also showing some good activity. And so the ruxolitinib is an anti-cancer and JKI inhibitor, and treatment of bone marrow cancer. And similarly, like, they have used thalidomide. Thalidomide is again an anti-cancer compound. It is already, as I given you, like, it is anti-inflammatory, but it has already it is used as an anti-cancer compound. And so these are a couple of molecules, and which are already, they are repurposing and checking some of the repurposing these molecules. And another is tofacitinib. Then all these molecules, once it comes up, but what next? So we need to think about some of the new innovative molecules as well. And so the research has to happen. But once we come across some of those studies, and we may have to change maybe some of these new molecules and bind with its either the spike protein and or the the SE inhibitors, and create the novel molecules. Otherwise, there is people are very hopeful, and scientific community is under so much stress to build the vaccines. Although the vaccines, once even if it is made, also to implement, to reach to the every human being, it will take a lot of time, but it will be done in a war footing, as the situation is not so easy. And hopefully, whatever I have put couple of slides in mostly focusing on why the direct drug discovery is not a feasible one at this point of time, but we can repurpose. We can take several molecules, we can check their efficacy, and utilize to control the pandemic. Then next, probably we have to look for either the antibody generation or administrating these antibodies to improve the health conditions of the people who are in severity. Similarly, the vaccine research, although it will take some time, it will not happen so soon, because there are the molecules are new. They need to go through the phase 1, phase 2, and phase 3 studies, and again, and there are issues, could be or no issues, and if we are lucky enough, we will get it much faster, and that will be a blessing from the for the human society. So basically, I have brought through a existing drug and utilizing this existing drug, repurposing, and maybe further research can be happen, but not immediate, but at least after we come through, we overcome this challenge, and after that probably we may have to think some more innovative anti means anti SARS against moieties, and we have to develop new molecules as well. So with this, actually, I would like to thank all of you, your time, and I look forward to any questions.
Yeah, thank you, Dr. Balram Petro. It was nice collection of information about the drugs to treat SARS-CoV-2. Professor Vijay, over to you.
Yeah. I have a question. It's excellent. Thank you very much for this enlightening talk, Dr. Patra. So, but I have a very simple question, a very simplistic attitude. See, in drug repurposing, there are two family of molecules. One is the chemical entities, wherein you can use the docking model simply and change the chemical functional groups in the improved binding if we know the target. Okay, if we know the target where it is going to bind, so we can, that is what some of the people are already using, taking the natural product and changing the chemistry of that product and repurposing it. Okay, but then now coming to the protein molecules, because there are a lot of protein antibodies, as you have mentioned yourself in your talk.
Yes.
They are repurposed because probably the pathway is the same. Like in some of those things that you talked, it is inflammatory pathway. So cytokines are probably not produced. So that is one thing. But in the what you call the regulatory point of view. Now coming to the regulatory point of view of repurposing the protein molecules. Can you say something about it? Because it's not the same regulatory aspect for the repurposing of antibody or antibody fragments compared to the other molecules. And what are the hurdles and how you can compare?
Yes.
Because many of these MABs and their fragments have been studied. Now repurposing is happening.
Yes.
So some companies are doing that. So can you comment on that?
Yes. Well, actually, like, I actually divide your question in almost like 3 to 4 different segments.
Okay.
One is like, the first one is repurposing the drug. That means, you can't touch any changes in this molecule. That means whatever, like, suppose ritonavir or could be our all these molecules which are utilized, for example, favipiravir or remdesivir, they are same as exactly as the molecule which is already existing in the market.
No, I do agree, but then you can modify and then repatent it, and that is not going through the same.
That's what I'm coming to, madam. Actually, see, the moment we do any changes, the moment, suppose, for example, we can put in a, if you really see this molecule, they have some similarity. Actually, if you take the favipiravir, it has not much similarity to the remdesivir. But for example, to the some of these acyclovir and remdesivir and all, they have some similarity. They have a nucleoside ring, and they have a 5-membered sugar ring, and they have an amino acid component. So basically, if you see all these changes, at least we can do a lot of research. But what I am trying to tell is actually like, what next, when I was talking, I think we have what next is these things can come up. We can do some modification. We can do some of the computational chemistry and see the drug-like properties of those molecules. Either could be virtually or could be really synthesizing them. So that can be the next parameter. But in the repurposing, we have to use exactly the same molecule.
Same function, okay.
Yes. So what you said is actually the antibodies and the large one. See, the God is great actually. When he gave us this issue, also he has given us a body defense mechanism. He has actually, like, we have the blessing from the God, like, to build inside the body to create an antibody defense mechanism which creates and people come out of this problem. That means, you see, we are not taking any kind of new molecule, and only thing is it may be not be same for each different user. Like, that's how we have to do an testing some of the feasibility for each antibody cannot be administered to everyone the same one. So it has to be do some of the doctors needs to test them and see the feasibility and utilize those antibodies where actually they can also inside our body, they can grow and they can create an antibody and create a defense mechanism against the Ebola or the SARs, and all these things will happen. So it will be also a kind of repurposing. Means, like, that means, we are utilizing the body's defense mechanism to create a positive movement in this treating the people. So that is our second things. The second thing may come up. The most other important thing you have asked is actually natural product based. See, even when I was hearing you in the last thing also, you were asking the same thing. I think that's actually natural products are not indifferent from the either it is a small organic molecule or could be in a large molecule, but they are chemical molecules. So as a organic chemist, I visualize they have the potential. Only thing, how do we administrate? How do we utilize them? How do we study their feedback and their mechanism of action is actually the difference we have to create at least as in India, I think the whatever we talk about Atmanirbhar Bharat, I think in the Atmanirbhar Bharat, actually, we have got the blessing of almost 1,000 years, our Ayurvedic history, the Charak Samhita, Susrut Samhita and all. And probably those either the know-hows must be utilized and utilized for the drug discovery process. At least the next generation of drugs should come from there actually. So if you ask me actually honestly, because you see, I'm not talking anything in air because it's already the drugs, which is paclitaxel. Paclitaxel is actually, it came from the
Zoom lo unnappudu actually video on cheste video కనబడుతుంది పక్కనే అది మైక్ ఉందండి అది
please mute yourself.
మ్యూట్ లా ఉంది దాంట్లో గ్రీన్ మటుకు కనబడుతుంది నాకు తర్వాత నేను సెలెక్ట్ చేస్తా ఉంటే సెలెక్ట్ మైక్రోఫోన్ లో దాంట్లో సేమ్ యాజ్ సిస్టమ్ తర్వాత మైక్రోఫోన్ రియల్ టెక్ ఆడియో దాన్ని టిక్ చేస్తే ఓకేనా అది వాళ్ళకి వినబడటం లేదంటున్నారు
Sir, we are able to hear you, sir.
Dr. Naidu, please mute yourself.
చేయమంటారా
No, it's not required, sir. We are able to hear you.
చేస్తేనే అవుతుంది కదా
వినిపిస్తుంది సార్ మాకు అందరికి మీరు చెప్పేదంతా వినిపిస్తుంది
Dr. Naidu has muted. Yeah, Dr. Naidu has muted. Yeah. Dr. Balram, sir, please proceed, sir.
So the like my perspective, like if I have enough money and good chemist, I think I will spend my time for next 10 years probably in the drug discovery in the natural products, because we have got from paclitaxel, we have got vincristin, which are novel anti-cancer compounds, and there are several of them. I don't need to
many molecules have come.
Yes. So I have answered actually, madam, in your question actually in 3 phases, and I think I have answered to all your questions.
Dr. Balram, Dr. Dinkar Sahal wants to ask you a question. Who is one of the panelists?
Yes,
Dr. Dinkar Sahal, please.
Yeah, I have heard from Dr. Balram as well as Dr. Srinivasan that thalidomide is being considered for repurposing. But thalidomide is an optically active molecule, and my question is, which of the 2 enantiomers is being considered for repurposing?
Actually, sir,
I am audible now.
Yeah.
Sir, I am audible, Dr. Naidu.
Yeah, Dr. Naidu, you are audible, sir. You are audible, sir. Yeah, please mute yourself,
thank
sir.
You very much, sir, because the mic was off.
Yeah, okay, sir.
Okay, right. Thank you very much. Thank you very much.
Welcome, sir. Welcome. Yeah, Dr. Dinkar Sahal, please, sir.
Yeah, so I think Sir has asked me in the question, which is the isomer is the active?
Yeah, thalidomide.
Yeah, thalidomide. Actually, like sir, thalidomide, whatever the structures they have given, they have not given any the chiral molecule. They have given the mostly the but as actually I think must be
maybe chiral mixture.
Yeah, there must be we have to check that details, sir. Actually, I don't.
Right.
Yeah.
Right. That's a good question, sir. So there's a question from an attendee, from the participant.
Yes.
Is docking also used for identifying immunomodulatory drugs?
Yeah.
Yes.
Right.
Yeah.
And between which molecules is docking done? Because the target of these drugs is not pathogen. That's the question from a participant.
Okay.
Yeah, that's what my question also, part of my question.
Actually, they normally, if you know the target, then only we can take the target, because normally they bind the target with the molecule. So otherwise, sometimes they do some of the other methods, again, to identify drug-like properties. But most importantly, in the either it is a CADD approach, computer aided, they need a target, and without the target you will not be able to do any binding assay. So that's why it is important to have the target.
Right. Okay, sir. One last question.
Yes.
From the participant. Artificial liver and kidney are available now. Are they usable for preclinical toxicity studies?
No, I've read also the the next generation. Actually, the artificial kidney. I think it is an inventor is actually an American Indian, and actually we are hopeful. I think once but it has not come to at least to implementation yet. But I think if once it comes, I think it will create. I think mankind also is very creative, very creative. Although the diseases are also become very difficult, but we have to coexist, and we have to.
That's preclinical.
Yes, we have
Yes,
to.
sir. Here I would like to say one more thing, sir. Exactly.
Yes.
Now there is what is known as actually organ on chip. And there are very good experimentation where actually on chip you can have a kidney and lot of kidney function can be evaluated.
Yes.
Right.
A lot of study has been shown that many of the drugs, those who are actually nephrotoxic, we know they have shown that yes, they are showing. So organ on chip is becoming one of the very popular method
Yes.
for toxicity study.
So
and almost every organ on actually kidney, their lung, their all those things are working very well for very initial screening of compound.
Maybe to some extent it is helping in the stem cell research. Actually, it is helping to create a new new artificial body parts. Actually.
Okay. Okay, sir. I think there are no more questions now. So thank you very much for accepting the invitation and giving a nice overview of all the drugs that can be attempted for SARS-CoV-2. Now over to Professor Krishnan.
So thank you, Dr. Balram Patro. So it was a wonderful lecture. I would like to I take this opportunity to thank all the speakers, Professor Srinivasan, Professor Dr. Rishi Jain, Dr. Balram Patro for giving a wonderful insights into the drug discovery programs and also repurposing of drugs, and to a small extent on the discussions were also focused on natural products. So we hope to hear more from other speakers as well. Thank you all very much. I also take this opportunity to thank all the participants who participated in this session. So now we break for lunch. We assemble back here by 2:30. We are going to start the second session. Dr. Naidu will start his lecture from 2:30. We'll continue from there. Thank you all so much. Thank you,
sir. Thank you, sir.
I request all the panel members to be back by 2:15, 10-15 minutes earlier. Thank you.
Thank you, and thanks to all of you, sir.
Thank you, sir. Thank you.
Very good talk. Yeah.
So we'll start at 2:15, ma'am. We'll start at
Yeah. Okay, fine, no problem.
Yeah. I'm waiting for the polling to finish so that I will.
Okay. Okay. Thank you.
Okay. Thank you. Yeah.